Skal vi teste lyden? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
Hello? Okay, welcome everybody. This is the third time we are arranging this Living Norway Colloquium. And we are 80 people attending this workshop from 45 about in this room. Is an echo here? Yeah. Hello? The echo. Should I keep calling, talking? Er lyden på PC på? Kan vi se. Hører dere? Nå er det borte nå? For nå slo jeg av lyden på PC. Okay. Okay, welcome everybody. This is the third time we're having this Living Norway Colloquium. We are having 45 people attending here in this auditorium. And about the other ones, altogether 80, are attending abroad. We are attendances from 11 different countries abroad. It's still the echo. Hello? I'll just talk slowly. It's as you understand, it's a combined virtual and a live conference. This year our theme is the ethics and technical know-how of how open science in ecology and evolution. Before I start, I'll say something about NINA. We are the Norwegian Institute of Nature Research. Our vision is to contribute to sustainable development with research and knowledge on biodiversity and climate and society. We are an independent private research institute, non-profit. We do applied and basic research. We are having 300 employees, and we are the biggest research institute in Norway that are working with biodiversity and the society. We are having an interdisciplinary staff with both ecologists and social scientists and humanitarians. We are all working with biodiversity in one way or the other. 80% of our scientific staff are having a PhD. Here you can see the picture of our main headquarters, which is here at this house. But we also have local offices in Tromsø, Bergen, Lillehammer and Oslo. We are, as I said, our fundamentals are our knowledge of species and ecosystems and the society at large. This basic knowledge we use to have different focus in our research. We do mapping, monitoring and gathering environmental data. We are having big research effort on ecological condition and ecosystem accounting, nature and climate change, restoration and nature-based solutions, ecosystem services, environmental sustainable society and businesses, and technology and innovation. As you all know, we are having a really big challenge. We have the biodiversity crisis and the climate crisis. I think we all here attending this workshop should contribute to a more sustainable Earth. The Living Norway will contribute to mobilize fair data and tools to interpret data. Education is in focus. So as this big crisis is coming, we need to put data together in a new way to get really good analysis. The Living Norway is, as you know, consisting of 9 different Norwegian institutions. And we have this year established an interim board, which I am leading. The Norwegian Research Council has this year found this really important network, and they have funded this conference. So we are very pleased to be here today. And I wish all of you a fruitful colloquium. I think this is really important. Lykke til.
Hello? Is this on? Can you hear me? Yeah. And no echo this time. Okay, perfect. I don't know why that was happening, but if someone are having their Slack or the YouTube stream open, I suggest you close that. That might be one of the reasons why we were hearing this echo. Maybe, I don't know. Let's hope it works well for the rest of the day. Yeah, I will mute this for 2 seconds when I find the slides for the next presenters, which is me and Anders. Yes, we are a little bit ahead of time, so I will start with some practical information. Signe has already wished you all welcome, so I will just reiterate that. It's great to see you all again. Many of you came also for our colloquium last year, and some of you are new to this event, and we are happy to welcome you all. We also have an unknown number of online participants, and probably some that did not sign in but are watching the live stream. So I hope you also enjoy this the session we are hosting. A few words, sort of practicalities. First, I will be the session chair of this first session. I will also present, so when I I'm giving my presentation, there will be no one to help me keep me on time. So I take a big responsibility when doing it like that. In the case of emergency, at least those that are present here, those that are online, I don't know, but those that are present here, in case of emergency, just follow the green exit signs in a calm and well-behaved way, and then we meet outside the building after. And then we will get further instructions. There will be a lunch at 12 o'clock. If there are anyone in the room that did not sign up for lunch, it should probably still be possible to join, but please let us know as soon as possible. Let me or Annette know. It should probably be possible to solve that. The same is the case for the dinner tonight. Hopefully those that are planned to come have signed up, but if there are anyone here that want to join but did not sign up so far, it might still be possible to join. Annette, isn't that true? Yeah, but please let us know as soon as possible so that we can let the cantina know how many will be there in the end. Finally, I will say a few words about the Slack. I will present a little bit more later about how we are going to use it in the conference, but I know that there are some people that have difficulties logging into the Slack work space that we are using for this conference. I had some problems myself when I started and got some help to solve it. If you are having problems with that still, please contact Matt Granger. He's sitting there waving his hand. If you are online and still also have problems, send an email to Matt. Matt Matthew.granger@nina. Matthew.granger@nina.no. Or you can contact him through Twitter or somehow. Get in touch with Matt. He will help you. Yeah, I think that was the practicalities, and we are ready to start the talk in 10 minutes. No, 10 seconds. Yes, so then we are ready to start the first presentation of today after Signe's welcome. My name is Erlend Nilsen. I'm the PI of the Living Norway Ecological Data Network. I'm also a senior researcher or senior research scientist here at NINA. And I have a part-time position as professor in ecology at Nord University in Steinkjer. I will do this presentation together with my co-PI, Anders Finstad from NTNU Museum, which is a professor in biodiversity. So I will give a very brief introduction first, and then Anders will tell us more about why we need Living Norway, and then in the end I will tell a little bit more in detail what we are doing. If you want to tweet about this event, then the hash tag Living Norway 2021 is sort of recognizing this event. So, what will we address at this colloquium? Of course, we have a title that tells us about the main topics of this colloquium. But there is also a sort of a broader picture here that was already mentioned by Signe, which really motivates why this is the work we are doing here is important. As we all know, and that has been documented in several reports, biodiversity is going lost and ecosystems are becoming degraded currently, in large part due to human activities. Ecological research and other research, but since we are ecologists, we will focus on that, based on empirical data, is really needed to tackle this crisis. Of course, we need to develop theories, hypotheses, and so on, but we really need empirical data collected in the field and sometimes in the laboratories to test these ideas, to test these hypotheses, and to confirm them or reject them. And since we are dealing with issues that have a really huge societal impact and are of huge societal concern, it's really important that this research that we are providing and conducting, it must not only be excellent, but it must also stand up to the highest standards in terms of reproducibility, openness, and transparency. And these are the things that we will mainly focus on in this conference. Our today research we do conduct in different fields of ecology and beyond, but this last part here is common for all of us. Few words about the program. I hope you have had a quick look at the program, but you know that there are 3 main sessions today. In the first one, which is the title, What Tools and Know-how Do We Need to Manage Our Data According to Modern Expectations? It's this session, and we will give short presentations about new tools and new findings that are relevant for modern data management. Then we will have lunch, and after lunch we will have two sessions. The first session after lunch. Sorry about this, but the text on the screen was so small I can't read it. We'll focus on ethics and best principles in a fair and open data landscape. Then we will have a short break, and the third session will be user perspectives on open data and open science in ecology and evolution. Tomorrow we will have a bit of a different type of program. Before lunch tomorrow we will have a workshop where we will present and discuss and further develop novel tools for reproducible, transparent workflows in ecology and evolution. And this will be a bit more hands-on than today. So today is mainly lectures, talks, some discussions. Tomorrow is more workshop style. Then after lunch tomorrow, we will have something that we call open work meeting in the Living Norway Steering Committee. Maybe we should have used a different title, and now we have on the slide a different title. So basically we invite all of you to join for a work meeting where we discuss further our plans for 2022 to be added in our work program, and we have some specific points that we would like to discuss. I will say a little bit more about that after Anders. Yes, I encourage you to have a look at the conference guide that is posted on our webpage, and it's also, you can find it in Slack. There you find the description of how we intend to use Slack during this workshop, and there is a description of the code of conduct that you should have a look at, so that we all behave well to each other during this event. I will not say much about how to use Slack, but the main thing is that we encourage you, both those that are joining online and those that are here in the auditorium, to ask questions in Slack. And then the moderators will pick a few of them to ask to the presenters. That's how we sort of organize the discussions after the talks. So please, also you in the audience here, use Slack when you ask questions. You find the right channel, and in there you will raise your questions. Okay, before I give the word to Anders, I would just like to remind us that during this biodiversity crisis, ecology is really a central science that we need to let the society around us know that we are here to solve important questions that could contribute to solve the biodiversity crisis. So with that, I give the word to Anders.
Thank you. And still got an echo here, or no? Some better? Yeah? Or is it just when I try to close onto this, there's some microphone on here? It's better, but no, that was not better. I think we do that into.
Yeah. That's on? Yeah. No, I'll do that. That's better. Yes. So what I really wanted to take the opportunity is to start reminding you what we are talking about. And as you might have noticed, the sustainability discourse is pretty high on the agenda for research and development and the society at large at the moment. Now, I like this representation of the sustainability goals. It's from the Stockholm Resilience Center, published a few years ago, and it's pinning up to the definition of sustainable development as that meets the needs, meets the present while safeguarding the Earth's life support system. And the Earth's life support system, that is represented here by the 4 Sustainable Development Goals, that is life in land and water, freshwater, and climate. What most of those working in our field knows is, of course, that this hangs on an interaction between the living and the abiotic factors. This is what makes up the fundament here. And what other names we have for that interaction, that's ecology. So ecology is not only fundamental, it's also, and biodiversity is not only fundamental, it's also very high up on the political agenda at the moment. Most of you might have recognized that There are going on high-level international discussions on biodiversity. The nature crisis, as Signe mentioned here, is being fronted. I think still we might do a better job on promoting the nature crisis versus the climate crisis. It's a bit in the shadows, and still it's a bit confusing to the general public, at least, which is what here. That's a digression. My main point here is to say that, well, we have politicians, decisions makers, all rely on information that are given to them by science and the scientific community at large. And this goes out from the small one, which we have on the right-hand side here, from local area investigations over towards more national scale, towards the global scales, even from organizations like IPBES and their indicators to the planetary boundaries indicators. Trouble is, do we really know that all of the information and all of the knowledge that's generated here meets the standards that we have for good reproduction, reproducibility, and transparency in science? Are we up to the standards of open science in this work? My answer is no. I think it's obviously, and there are several reasons for that. So I'll put out 3 villains in this regard. Sorry, no good guys here. But I think that is the data, it's the workflows, and it's also the results that are currently not reproducible, not as open, not as accessible as we would like. So first of all, the lack of empirical data. That is really a no-brainer. We don't even know what species we have on this planet, and let alone how to protect them. I'll say a few words about that afterwards. But I do think that the lack of empirical data and how we're going to address these large empirical knowledge gaps in the future also hangs on point 2, which is lack and access to methodological details and raw data and research materials, because if you don't know what you have, it's very hard to plan what to get. So in order to make the knowledge gaps, we need to have open data to prioritize what is the most important things to investigate. And of course, there are complex workflows, complex data sets, increasingly so, and we need to be better of handling that. So, data gaps. I just briefly want to show you this. This is work done by Ivar Herfindal at CBD. It's the municipalities of Norway. Each bar is a municipality. The top bar here is the number of observations, and the left bar is the density of biodiversity observations, and it's sorted by various counties. So the main point is, for the most of the municipalities in Norway, we actually do have precious little biodiversity information per se. Same for the taxonomics bias. This is also well known. For those who don't read Norwegian very well, you can say that the biggest bar here that outruns everything is birds, fugler, and most of that is citizen science observations, which is the blue bars, and other professional observations are the red bars. Now, that being said, given that biases we have, the vast majority we have of the open biodiversity data are still occurrence only or presence only data, as we call it. Means it's information contained there, is the place and the time and the organism name. The more complex data types are lagging behind. We do have some of the technicalities for publishing this in what we call the event type of data, but it's still lagging severely behind in terms of getting out. Probably and this is my guess again, is the time, resources, and willingness to share, which is central, or most central. Also, workflows. As I said, we do have increasingly complex workflows. We need to learn to work with them as individuals and as a community of how to actually deal with data. This is a result of both more complex data sets and more complex advanced statistical tools that are used in mainstream ecology today. So, but for these workflows to work, they need data. And I would say they need, in order to be transparent and open, they would need fair data, which is basically short for machine readable, open, findable data. They don't need to be open to be fair, but at least it's machine readable, so they can feed it into workflows. What's the main reason we're lacking this? I are under the impression that this is mainly due to resources and motivation for getting data out there. So it's resource demanding, definitely, but we do not have the initiatives. For example, raw data, unstandardized, should be pretty simple to publish and make open. This is data repositories as we know it today. The technology is there. We just don't do it. Why? We tried to address this, me and Erlend, in a few years back. So we had a little sort of flashback. So in 2015, we wrote an opinion piece at forskning.no where we asked, why is not the data out there? So research data and public funded data is not shared within our field. There is a strong initiative for privatizing data because that is giving you authorship, which is on publications in the end, which is what gets you a job and which gets you new research funding. But there are precious little initiatives for sharing. So we got a prompt reply from the Research Council, which said, oh yes, of course, we need to do it. This needs to be done and dealt with. We need to do something about this, and we need to do it now. Trouble is that we're still in 2021, and actually precious little has happened except a lot of paperwork, a lot of reports saying how important this is and that you should do it. And rounding off, why do I think this is really important that we address? I think it's important since the progress on what we do and everything is relying on public trust in science. We do know from the big sort of past type of climate science type of what they call the climate wars, and also past in the 70s from the tobacco industry, that there are forces that are out there and want to undermine that public trust in science. And we need to meet that, and you need to meet that by doing transparent and reproducible research. We have sort of a taste of that going on in our own field. Everyone working with large carnivores or with aquaculture has known that there are forces trying to undermine the science. And that's my opinion, so that's it. And it's important that this is addressed. And living in Norway and the techniques and what we front there in terms of open data is, in my opinion, an important way to do so. And with that, I'll leave the word to Erlend.
Yeah, as I feared, we are using too much time and talking too much. So I will go relatively quickly through this, but it has to be said before Chloe gets the next speaker, gets the microphone. Yeah, so a few slides about Living Norway, who we are and what we are up to. Basically, we are working with open data, data infrastructure, educations, network, and to change culture. But the main objective is to make accessible, standardized, and documented data from field-based ecological research and monitoring. And as Anders also said, we are becoming increasingly interested in all the things that are surrounding this landscape, the open science landscape. We are having activities along a set of different axes, including education, networking, building data infrastructure, and so on. We are very tightly connected to the Global Biodiversity Information Facility, GBIF. We have a very good collaboration with the Norwegian node, and we have good collaboration with the international and the Secretariat in Copenhagen. And importantly, we build on the same standards and the same underlying infrastructure as GBIF. So it's sort of an extension into the ecological world. That's how we see it. We think that to make your data really fair, we should not just focus on the technical standards and the metadata standards that people often do, but we need to also focus on standardizing the data themselves, and that's where the Darwin Core enters. Some of the current activities. We work to build tools for data mapping, documentation, visualization, and management. This is partly or mainly involved building our tools that will be presented later today. We are working on a data portal and planning some archiving services. And the Living Norway data portal is part of the GBIF hosted portal program, so we are working together with them to achieve that. We are also involved in best practice or developing best practice guidelines for the use of data standards in ecology. So not inventing new standards, but working together with the international community to implement and further develop standards if needed. And we have quite a lot of activities related to networking, outreach, and coordination of various activities, including workshops and this annual colloquium, presentations to various groups are in Norway and outside. We're also about to launch what we call an open science lab that we will discuss further tomorrow. So we hope you will join us on that, because that will be a lab that is more open to the whole ecological research community. Yeah. So a few words about the discussions after lunch tomorrow. I hope all of you will join us for this work meeting. 3 points that we will for sure discuss. It's the Living Norway data portal. It's extending the Living Norway R package that Matt will present later today. And it's the Living Norway Open Science Lab. If you have other suggestions for topics that we should discuss, please let us know. And also, if you want to join us online, please let us know so we can arrange that. We did not set that up yet, but that it's easier to do. So, before I leave the microphone to Chloe, I would just like to thank those that have funded this event, which is Nina and the Research Council of Norway, and then in-kind contributions from all the network partners that are listed down there. I don't think we have time for questions to the first presentation, so I think we end it there, and Chloe is next presenter.
Okay. Does this work? Yes. Okay, so Alan and Anders had a bit of overlap with me in terms of time, but there's also a bit of overlap in terms of content, so maybe I can do a few things with a bit less detail. So my name is Chloe, and I have recently started as a researcher at NINA, but I've been involved with Living Norway further back than that, and open data and open workflows have been on my mind for a while. But what I'm going to talk about today is more motivated with where I come from, at least academically. Because I'm a population ecologist by training, and I kind of grew up in an environment surrounded by giants. Giants in the field that own and manage and administer data that is just big, big treasure troves. So I'm going to talk about gold standards for expensive data. But first, I just want to say that at least I think that, like the others already said, open science is crucial to tackling both the biodiversity and the reproducibility crisis. And just looking at what has happened over the past few years, actually quite a lot of good has already happened. The community has undertaken some large efforts to address challenges, and there have been some true successes. So there is still much to do though, and today's talk is about two things. And first, it's about data, because the open data practices and fair data practices have not permeated equally into all fields of ecology. And I'll be talking a little bit more about what exactly has happened or not happened when it comes to those really valuable data. And then the second part of the presentation is my own short introduction on what goes beyond the data, and essentially what is one of the large topics at this year's Living Norway Colloquium, and that's standardizing not just data, but entire research workflows. So first, I'm going to talk about developing standards for long-term individual-based data. But what exactly is individual-based data? Well, as the name implies, it's data collected at the level of individual organisms. And importantly, this happens not just once, but repeatedly over the course of the organism's lifetime. This means that if we want to collect this type of data, we need to be able to recognize individual organisms in the wild over time. So they need some kind of unique mark for each individual. And traditionally, people have been using artificial marks that are attached to captured individuals, such as bird rings or ear tags. And just as a side note for all the plant people, it's been done on plants as well. However, the problem with artificial marks is that you need to capture and handle the individual every single time you need to identify it. So not just once to put the mark, but also again and again to read the mark. And it's very work intensive and can be really problematic and basically impossible for some elusive and highly mobile species. So there's been a lot of innovation on how to deal with that. And one type has been to use natural marks instead. So no capture required if you can recognize a whale by the shape of its fin, or the leopard by how the spots are distributed. Or even better, if you can just collect a hair sample and identify the individual by its DNA. If you can capture it once but want more updates, you can also work with transmitting devices, such as the kind of passive transponder tags often used for studying fish, or active transponders, transmitters like satellite collars. What hasn't changed, though, is that irrespective of the method, identifying individuals and obtaining data on them is still very expensive. It's a lot of effort, both in terms of work, money, and sometimes logistic challenges. So these are some of the examples of species for which individual-based data has been collected for many years. And I'm quite sure that many ecologists and evolutionary biologists will recognize at least some of them. That's because some of these larger data sets are actually very famous. And the reason is that all the money and effort spent in collecting this data really pays off. So usually a single long-term individual-based data set can result in several dozens of publications. And these kind of data have really been the core, the center of the study of population and evolutionary dynamics and how they respond to environmental change. So exactly the type of knowledge we need now to address the biodiversity crisis. So some of the data sets have resulted in over 100 publications, and that was by 2010. And it's also quite well known that the longer data collection goes on, the more publications are produced, and also the more, the higher profile publications are produced. It also reflects in how relevant the obtained knowledge is for policy making. And here too we say the same pattern, and that is that long-term studies are really crucial. So these data are costly to collect, but they're a veritable treasure trove. Recently, there's been more recognition that this type of data is becoming so much more important, not just for more single species studies, but for larger scale and integrative studies. There's a problem though, and the problem is that for every single one of those several dozens of publications, there is a published data set somewhere, or more specifically, a fragment of the actual data set. Usually these fragment data sets are deposited because journals have policies requiring data deposition when they're going to publish an article. And these submissions, they usually just about satisfy this requirement, and the requirement being data is available. But usually not much more, meaning that we have a lot of fragment data sets that are incomplete, poorly documented, and disconnected from each other and from the source data. This happens both accidentally, just because, and sometimes intentionally. And it reflects a little bit that there has been some reluctance towards adopting open and fair data practices among the owners of such long-term individual-based data. And there are a variety of reasons for that, and I'm not going to give a comprehensive overview over these now. But instead, I want to focus on one specific barrier to making long-term data fair here. And that's the workload placed on individual researchers. This is very often a reason we get why data is not made available or not shared. People don't have time for this being documenting, reformatting, and so on, basically getting their data ready for being deposited and or shared. So if we want more data to be made fair, and especially large data sets to be made fair, one important step is reducing the workload of individuals. We already know that this can have a huge effect in practice, also with people working on long-term individual-based data. And the example I'm going to use is the example of the recently founded Spybird's Data Hub and Network. And it's a network for people collecting data on birds. And essentially, what the network does is it provides a service where they take people's monitoring data in whatever format it may be, and then builds reproducible data pipelines that convert it into a fair standard format that the community has agreed upon. And the uptake has really been very good over the last 2 years that the network has been running. And a lot of people have joined up and agreed to have their data made fair. So if you take the bulk of the workload away from people, suddenly they're much more eager to be part of this brave new world. Ideally, though, we'd have a standard format that doesn't just work for birds. because we have long-term individual-based data on all sorts of different species. So ideally, we have a standard format that works for all species and all means of data collection without losing any information. It should also be ready to share and compatible with large biodiversity databases. And also, ideally, we can integrate it very easily, both with other datasets of the same type and with datasets of a different type. Sounds like a pretty heavy list of requirements, but the good news is we already have a standard that fits all those requirements and is ready to be adapted to this kind of individual-based data, and that's the Darwin Core. The standard is designed to facilitate sharing and combining of information about biodiversity, and it's already commonly used for many other types of data, such as occurrence data. And it's also what is required by some of the larger biodiversity databases like GBIF. It uses a hierarchical structuring, which means that there are nested data tables containing different types and different levels of information, and that really makes it versatile enough to capture the complexities also of individual-based data. So as you can see, the advertisement line here is that Darwin Core is the way to go for distributing and sharing your data, and also for storing and archiving it on different platforms. However, I started this talk by saying that it's very important to keep the users in mind. And what is best for archiving and distribution may not necessarily be best for people both on the first end of the part, namely the data owners that enter the data into their databases, and on the other end for the people that analyze the data with all kinds of modeling tools. So both the data owners and the data users are quite used to working with a single table longitudinal data format. And that's not something that will just change overnight. And we don't want to make more work for people, so we really need to use the Darwin Core in a way that makes it possible for people to still access their data in the format they're used to. And that's why I like to think about the Darwin Core as kind of an envelope in which to pack and wrap your data for distributing and storing, and that can then again be unwrapped to use it. The actual process of wrapping and unwrapping can be quite technical. And again, we don't want to put that workload onto the already overworked researchers that own the data. So the key here really is that we have our tools ready that facilitate the wrapping and unwrapping both ways. If we can manage to get that going, that I think is really the way that everyone can start standardizing and mobilizing their own data much more easily than today. So next, I'm just going to briefly talk about workflows. And so far, it's all been about data collection, standardization, archiving, sharing. But we all know that scientific workflows are so much more than just raw data. It's been a bit similar with the whole open science movement, insofar that the development of standards and best practices has had a very heavy focus on the data side of things. That's extremely important, but what about the rest of the scientific workflow? How are we doing with the rest? And the answer is we haven't done an awful lot yet. So there's a lot to do to develop standards, not just for our data, but also for our workflows so that they can be accessible and reproducible. So I'll illustrate this a little bit with an example on integrated data analysis. So integrated modeling has really taken off. Excuse me, this is an old slide there. Integrated modeling has really taken off because it allows to combine different sources of data to get inference that wasn't otherwise possible. And the way this is done today is that I take my data A in a random format and my data B in some random format, and I do something with it, and then I run an integrated model. And some people provide code, some people don't. Maybe there is some documentation somewhere, maybe it's useless. It's really up to the individual researcher how this looks. And then there's some very specific inference. And in the kind of studies that come from individual-based data, it's typically about a single population of a single species in a single location. And let's be honest, if we're thinking about the biodiversity crisis, that's not very useful anymore. What we need is analysis that are on a much larger scale and have a comparative element to them. So the situation's changing now because data is starting to be standardized. And as soon as data is standardized, the rest of the workflow can also be standardized. So for the workflow of an integrated analysis, we now start with two sets of data that follow the same standard format. They can be converted into a model input data and then fed into a typical workflow for integrated analysis that has a modeling step, some assessment of performance and fit of the models, sometimes follow-up analysis, and most importantly, it can give us results that are more general and more useful. So the crucial thing here is now that we need to fix this part. And this part is that all of the code that goes into the workflow should be published and properly annotated. And there has to be documentation that is sufficient for people to both understand, reuse, and adapt the methodology that is used. And it's really up to us now as the research community to work on standards for how to do and how to document our analysis and scientific workflows more generally. So I'm going to do the super quick summary of this presentation. And basically on the data side, there is a lot of very valuable data that is still locked up. And large workloads are one of the main barriers here. So if we develop good tools for reducing workload of making data fair, we will really make a difference here. And maybe some of you are thinking, well, that sounds very vague, so I'm going to refer you on to the next talk, and Matt and Joe will make it a bit less vague for you. And on the second point, we haven't done an awful lot yet on standardizing our research workflows. And that's really where we have a lot to do. And again, more advertisement from me. I hope that a lot of you will come and join the workshop tomorrow so that we can discuss together on how to approach it and how to make steps forward here. Thank you.
Oh, sorry. Yeah. So I will reiterate. Thank you, Chloe, for a wonderful presentation, and we will follow these discussions up tomorrow. And now, to try to keep the schedule, we will jump into the next presentation. And the next presenter will be Matt Granger, which is also a researcher here at NINA. The title is Living Norway R, Creating a Darwin Core Standard Compliant Data Archive, a data package for your biodiversity data. And he has worked closely together with Joe Chipperfield on this developing this package. So I'm looking forward to hear what you have to say, Matt.
Thank you very much. It's a Poison Chalice by Erland and by Chloe now. That's good to go. So I'm going to introduce you to the R package Living Norway R. And as Chloe mentioned before, we're trying to make a way to make it easier for researchers to share their data. The aims of the package are to create a workflow that's Darwin Core Standard compliant, ends up with a data package, it's an archive, facilitating fair data, sharing and uploading of Darwin Core archives to GBIF. So we're targeting GBIF, but actually you don't have to end up in GBIF. So you could just go to a point and stop. Also, we're providing tools for processing and manipulation of metadata. Now, some of you might remember me standing here last year saying how close we were to getting this package done. Yeah, well, it felt that at the time, but now, this year, here we are. We've got it done, but the main problem has been this metadata point, point number 3 there. I'm not going to talk too much about it today, but tomorrow we'll address it a bit more and how we've solved the problems. One of the feedbacks we got last year was there's some confusion, and I'm going to do exactly the same thing. I'm going to make this mistake between the Darwin Core Standard and a Darwin Core Archive. So, what is the Darwin Core Standard? It's basically it's a set of standards, set of terms that are controlled. So, when something in your data means something, then it also means something for everyone else's data. It's a standard. Those standards are really flexible and they're adaptable, which is really useful for ecological data. As an example, this morning I saw on Twitter that there was a discussion about dates and how America and Liberia are the only countries in the world to use the date format of the month first, 9/11, for example. But lots of people pointed out that there is an international standard for this. So you've got the year, the month, and the day. So in order to communicate with Americans, we can use that ISO standard about dates. So that's what we're talking about when we're talking about standards. It's allowing us to communicate between data sets, to integrate data sets, and that sort of thing. Ecologists often tell us that my data is unique. I'm an ecologist. I've said those words as too. So Joe's done us some nice slides here to sort of illustrate this point further. We have our 2 data sets. They're very dissimilar, very different. But when we talk about standardization, we're not saying we're going to put all this data together and make it the same. We're not going to hide those unique features in the data. What we're trying to do is to label those features in the data that are the same so we can integrate those data sources together. So we can have a better integration of data. We can use data together to make inference about biodiversity, sustainable development goals, all those things that you've heard from the talks before. Fundamentally, Living Norway R continues to aim to get us from here, which is a workflow that we often use. So you go out and you do your surveys. In the bottom there, I like PFAL, so we go and do some PFAL surveys in Thailand. I go to my office, I write up my data, do some analysis, then I write my paper, then I submit my paper, and the journal says, well, you've got to put your data somewhere. So after I've spent all the money, I've done all the fun stuff, then we've got to sit down and find ways to share my data. Okay? Often that's a difficult point to do that. So Living Norway R wants us to get from here to here, where the actual archiving of data, the sharing of data, happens right at the start. So you produce a data package, and you can use that for your analysis and for your write-up, and then you can publish it if you want to. This means that you're saving time, making it easier for researchers to share data in an easy, standardized way. Darwin Core archive files. So these are the core type of files that are used. They adhere to the Darwin Core standards, but they're a particular type of file. It's basically a zip file with a core data table. So typically this could be like an occurrence data table, so all the species occurrences you've got, or it could be an event table, so like a survey event that you've done. And around that core table, you have a number of extension tables. Okay, so those extensions could be things about genetics. It could be any kind of other thing. It could be an event table if your core is a occurrence table. Knitting all that together, you've got a metadata file that explains the data. It gives you a readable output. It can give you a readable output of what the data is, the title, who's made it, the abstract, where you go for it, get more information, all those sort of things you need, what type of data it is. And knitting all that together is a special file, that's the resource metadata, which tells you how to link together these different files. So that's the Darwin Core archive file, and that's what we're aiming to produce in Living Norway R, is this archive that is compliant to the Darwin Core standards. So I'm going to give you a rather ugly but rapid tour of the Living Norway package. You can go to GitHub now and you can download Living Norway R, and it should work. If you'd done this on Friday, it didn't work and I was in tears, but now, this morning, it does work, so we're very happy. Yeah, and if there's any problems you find, you can send us an email or put an issue on GitHub for us. Load the package, Living Norway R. And what we want to do is firstly, we're going to import a Darwin Core archive. So there's lots of Darwin Core archives out there. You can get them from GBIF, you can get them from Nina. This one is the TORV-E dataset, so bird dataset. We can pull this down, we can find it from the Nina server, and we can actually get this data into our package. And as I said before, these Darwin archive files have this core and extensions. Normally the core one is the one you want to look at first. So using some of our functionality, we can do things like get the core table and convert that into a data frame. And a data frame is something that if you use R, you'll know it's basically a data table, and you can actually use that to then, as you would any sort of data. Important point here is that we're using a thing called R6 classes, so object-oriented programming. So it might look a bit different to what you're used to, but what that allows you to do is build an object, so any sort of object like you'd have it normally in R, but then you can have specific functions that you tie it to. So here, that $ get core table, what that means is you're actually using that function for that object, the archive. Okay. So as I said, once you've exported it to a data frame, you can use it as you would any other data frame. And here I've just got some tidy code to link it up to a ggplot and then plot out a graph of some data. Okay, it's not very informative, but it shows the point that we can do this. We spoke about metadata earlier, and what we can do for the metadata is we can get the metadata out by using the get metadata function from the archive, and you can pull out bits of the metadata that's important to you, like the title, the abstract, so on and so forth. All these allow you to interact with these already made archives. But of course, you might want to create your own Darwin Core archive. And this is where the excitement bit comes in, the good fun. There's two different ways of doing this. For us, with this TORV-E event table, it's already in a format that allows you to automatically link up the two things because it's already been to Darwin Core archived. So you can do it automatically. You can automatically map it, meaning that you get the column names matched to the Darwin Core standard column names. So just illustration of what I mean, here we've got doing a manual mapping. We can show that we can link our column names in our data frame to the standard terms in the Darwin Core. So type is a type, modified is modified, so you can see that it's already been done, but this just illustrates the point here. with the Darwin Core archive, you have the core files, and you also have the extension files, and of course we can do the same thing with these extension files. We can bring them into R, or we can make them as well, and we can get them out as data frames. So there's full interaction there with these different formats, or different files, I suppose. The final step, if you're making your own archive, is to build the archive, and we can use different functions there to bring together different areas, different occurrence tables and event tables I've done here. I haven't added the metadata in to this object, but we'll discuss that tomorrow and how to do that in a way to make it a lot easier. But what it shows you is you end up with this, basically a zip file or a zippable file that is your Darwin Core archive. And eventually that can be automatically, but it isn't at the moment, put forward to GBIF or to a different server and then submitted there. Okay. So that's another step we need to take at the moment. So I know I've been really quick, and that's a quick whistle-stop tour of the package. But please join us tomorrow for the workshop, and Joe and I will walk you through the package in more detail. It's quite hard to give a talk about R code, but I'll try my best. But so tomorrow, it's really, if you want to come and join us, that's great. If you can't join us, go on to the GitHub site. There's the link there, Live in Norway R. You can download the package, you can play around with it, and at this stage, even though I'm quite fragile about the working of this whole thing at the moment, I think it's taken us a long time. Joe and I have had sleepless nights about it, but please come and break our code, okay? We're really keen to come for you to really test it and really try and break it, because we have quite we're all ecologists, we work in quite a unique area, quite a small area. We don't really work with genetic data, fish data, all kinds of different data that we need you to come and test the package with. And I'm guaranteed that as soon as you start this process, there'll be an error. It'll be an error that I have missed and not fixed yet. So please get in contact with us if you find an error. You can put an issue on GitHub if you use GitHub that way, or just send one of us an email, Erland as well, to give us what the error is, give us an example of how you tried to use it, what you expected to find, and what the error message was, and then we'll try our best to dig through the code and try and fix it for you. So just to reiterate, please join us tomorrow, and we can have a bit more detailed, hands-on play around with the package, and yeah, thank you very much.
Yes, thank you, Matt. I know a lot of people have been waiting for the package to be presented in the final form, and even though, as you pointed out, there might be some more testing might be needed before it's fully into production. I think this is really, really a great contribution that is made here. We have time for a few questions, if you are ready for a few questions, Matt. Can you elaborate a little bit about some of the functionality in the metadata part of the package? That's something we have been discussing a bit.
So yeah, as I said, the metadata has been a bit of a headache, something to sort out. We wanted to make it something that our users could use and make it easier and straightforward for them. So at the moment, the Darwin Core uses XML language to display the metadata. So what we're trying to do is to make a way from R to make functional XML, so machine readable language, but also we wanted to make it human readable. And that's been a massive challenge, and probably a bigger challenge than we thought it ever would be. So what we've done is to try and use R Markdown, which is a type of language that uses R, but also converts to different languages, such as HTML. We try to use that to allow the user a bit more freedom in how they set up the metadata. So you could actually, in theory, you could actually develop a data paper using our R Markdown templates to actually display the data, and you can run that, and hopefully it will change into this sort of it's EML rather than XML language, ecological metadata language, which will be GBIF compliant. So as I said, tomorrow we're going to have a bit more detail in how we've done that and how to actually get that working in your own archives.
Thank you, Matt. Yeah, I think we get ready for the next presenter. And the next presenter is Caitlin Mendeville. She's a PhD candidate at NTNU, and she will be talking about is the peer-reviewed literature a source or a sink for open primary biodiversity data? And this will be not fully, but at least partly based on a paper that Caitlin published earlier this year. So I'm looking forward to your talk, Caitlin. And I think you have to use that one. Be sure
Yeah.
you're okay with that.
Oh, yeah. It's kind of fun to hold the real microphone. Yeah, thanks. So, like Arlen said, the idea for this talk today is that I want to share a little bit of the research that is behind this systematic review that was published a few months ago, that I've been working on over the past couple of years with my co-authors listed here, who are all here today: Walter Koch, Arlen Nielsen, Anders Finstad. So the goal of this review was to dig into the peer-reviewed literature and think about its role in the sharing of open biodiversity data. We wanted to think about whether the literature tends to serve as a source of open data. Does it generate new open data or facilitate its reuse? Or does it tend to serve as a sink for open data, where data are reported in the literature, but then not made available for reuse? So I'd like to start by giving a little bit of kind of behind the scenes context to situate how I came to be interested in this work. So I've been working for like almost 8 years now in ecological citizen science, and I'm a really big proponent of the value that citizen science has to offer to expanding ecological data collection. So this offers huge benefits, of course, for ecological biodiversity research and conservation. But aside from that, it's also been well documented in the literature that participants in citizen science want their data to be used to make what they see as a positive change, to be used for research, to be used for conservation. So thinking about it from this perspective, I was kind of driven early in this project by this imperative to value the time and effort that citizen science participants put into gathering data, to make sure that those data are used to their full potential. And that often includes making sure that those data can be shared with the people who want to use them: biodiversity researchers, conservation practitioners. But as we started developing this project, it's of course clear that issues of data stewardship go beyond citizen science data. And a large part of that relates to the fact that it's increasingly hard to even differentiate citizen science data from other biodiversity data. Way back in 2014, Karen Cooper, a researcher, coined a term that I really like, describing the invisible prevalence of citizen science data. She was basically making the point that citizen science underlies a massive amount of ecological research, even when it's often not mentioned by name. As citizen science data become more ubiquitous, they're kind of just woven into the background of how science often happens. And this is especially true with presence-only biodiversity occurrence data, which is the data type most associated with digital citizen science platforms. So between citizen science and the digitization of museum and herbarium records, this presence-only occurrence biodiversity data has been just massively increasing in abundance and accessibility. We heard from I think all previous speakers today so far, how this occurrence data is one of the most easily standardized and shared data types. And you can really see that reflected in the growth of that type of data over the past couple of decades. So it became clear to us that we needed to look beyond citizen science, and we wanted to consider the role that the peer-reviewed literature plays in the open sharing of presence-only biodiversity data as a whole. Yeah, so as I mentioned, it's clear, it's well known and documented in the literature that applications of this open presence-only data have been increasing massively over the past couple of decades. But like in ecology, it's easier to detect presences than it is to detect absences. We realized as we were putting together our plan for this review that we knew a lot about instances where open biodiversity data are used in the literature, but we did not know a lot about how often biodiversity data are reported but not made open, and we didn't know a lot about cases where research could be using open biodiversity data but was failing or neglecting to do so. And we thought that kind of developing an understanding of these cases where data are not shared or open data are not used would help us better understand and identify barriers that are keeping these data from being shared and reused openly. So that was kind of the goal that led us into this review. To do that, of course, we had to look beyond open data infrastructure. So we moved to the peer-reviewed literature and undertook the systematic review of articles in Web of Science index journals that rely on presence-only biodiversity data. So our first question was just this: where do authors obtain presence-only biodiversity data? It turned out that 42% of articles in our review accessed some of their data from open biodiversity databases. But 81% of the articles in our review accessed some data from sources other than open biodiversity databases. So this was sort of the first surprise to me personally. A lot of presence-only data is being generated outside of this kind of well-known and quite talked about open data infrastructure that perpetuates this data type. For what it's worth, we also noticed that about 33% of these studies cited citizen science as the direct source of their data. So it is quite a lot of citizen science data represented here in this figure. Yeah, so looking at these initial results led us to some follow-up questions, and I'm going to talk about kind of our findings in three main questions for the rest of this talk. So first of all, we wanted to learn a little bit more about that 42% of studies in our review that use data from open databases. When these studies access and report on this data, how easy do they make it for other people to retrace their tracks and reuse that same data again? So we found kind of mixed results here. So we found out that of all of the studies we looked at that used data from open databases, only 7% of them cited a DOI or a URL that led to the specific database that we used. So that was quite low. We did see some bright spots though, kind of signs of optimism. So we saw that quite a lot of authors who didn't cite a DOI or a URL to a specific data set still made some attempts to credit the original creators of their data, for example, by listing all of them by name in an appendix. So we saw what we interpreted as kind of signs of data users trying to get there, but not quite achieving the best practices that are being recommended. And we think that some of this inconsistency probably comes down to the really wide range of open databases where users are accessing these data types. Articles in our study cited 117 distinct open databases, and many of them are quite small and targeted towards sort of specific geographic regions or taxonomic groups. So I think this is something interesting for the open data community to kind of keep in mind. While of course big data aggregators are a massive game changer and produce data that's used in thousands of studies every year, there are a lot of researchers out there who are relying on kind of smaller, maybe less well-supported databases. So it's, I think, important to account for that into the future and make sure that that data isn't lost. All right, so moving on from those studies that used open data, we wanted to spend most of our time thinking about the studies that were accessing data from other sources, this 81% of studies that collected some data from places other than open databases. So our first question was just, why? Why are these studies collecting data from places other than open databases? Currently on GBIF, there's nearly 2 billion data points, so does this mean that researchers are just not aware that they can access those? Does it mean that they're not finding the data that they need? We tried to dig into that question a little bit. So yeah, our first thought was that potentially researchers don't have the knowledge or the capacity to access open data in some cases. We couldn't really dig into this in our data, but it's been reported in the literature and is probably playing some small role in the results that we're seeing here. But we could find a little bit more evidence to support our kind of second hypothesis, that potentially we're seeing a lot of researchers collecting new presence-only data because the data they need aren't openly available. And this corresponds to a lot of research that's been conducted on spatial and taxonomic gaps in open biodiversity data. So one example that we found in our review is that the papers in our review looked at vertebrates a lot more often than papers in other reviews that were more focused on open biodiversity data. And this came largely from studies that were looking at mammals. So interestingly, the studies that were looking at mammals in our review were mainly getting their data from government agencies or originally collected data, not from open databases. And we weren't so surprised by that because mammal data actually makes up a pretty small percentage of open data. So this is kind of a sign here that researchers are collecting data to fill gaps where open data aren't available, which makes it extra important, right, that we're making sure that these data end up published so that other researchers can reuse them. That brings us to the last question I want to talk about here. We wanted to look at whether these newly generated presence-only data being reported in the literature are then being openly shared so that other researchers can use them. We found out that that is happening less often than we might have thought it would happen. A pretty small percentage of newly reported presence-only data among the articles in our review actually gets shared openly, and an even smaller percentage gets shared openly in accordance with FAIR data standards. So these results were pretty consistent among different characteristics of data, taxonomic group, region of study, and so on, but they did differ according to the original source that the researcher accessed the data from. Original data, collected by the study authors, was shared the most frequently, though still pretty low at a sharing rate of 27%. I think these results really highlight that in the conversation of opening data, it's important to be talking about both data publication, the new data being collected, originally by the study authors, and also data mobilization. What we see here is a lot of researchers who are gathering data from government agencies, private organizations. They might not, as individuals, have permission or perceive that they have permission to share those data, but working at an institutional level to help make these pools of data more openly available will reduce the effort in the long run that researchers are going to access these data anew each time they need them. So then finally, we wanted to think a little bit about what other barriers might be keeping the authors in the papers we reviewed from sharing their data. We're far from the first people to have thought about this issue. So the way that we approached this was to think about some barriers that have been proposed and discussed in the literature and look for evidence of those in the articles that we reviewed. So first, we expected to find a negative relationship between dataset complexity and data sharing, but we actually did not see this trend. We didn't find any evidence of a relationship between data sharing rates and either the number of original sources of data accessed by authors or the availability of metadata for those sources. We also expected that researchers who were combining open data with data access from other sources might be more likely to then share the data that was not open to begin with because of this demonstrated familiarity with open science. But again, we didn't really see any evidence of this trend. We didn't test this next one super directly in our data, but I think that the fact that data sharing rates are the lowest for sources of data that might have data ownership issues like private organizations and government agencies hints at the fact that data ownership and sensitivity are definitely barriers that prevent some authors from sharing their data. But lastly, I think it's super interesting that even in studies that we reviewed where none of these other barriers are really an issue, relatively straightforward data sets, researchers who are familiar with open science, even then, the data sharing rates were pretty low, which suggests to me that researcher incentive and maybe technical capacity, all of which has been talked about by previous speakers today, are definitely still major barriers preventing the sharing of open data. All that said, we did find some signs for optimism in our review. So you can see that the proportion of papers that share data is definitely notably increasing over the last few years. This top line here going down pretty sharply indicates data that is not published at all after being analyzed in the literature. And you can see that line is definitely in a downward trend. Yeah, so as I've been thinking about ways to reduce barriers to open data sharing, one thing that's really stood out to me from the papers that I cited in this review is this idea of the importance of taking a welcoming rather than a critical posture towards those who are taking their first steps into the open science and open data community. And I can attest to this personally, right? I introduced myself in this talk as an ecologist, as a citizen science practitioner. You might notice that I didn't lead with terms like data scientist or tech expert. So when I started at NTNU, I was pretty new to the open science landscape myself. And I still have a lot to learn, but I have to say, it's been super nice to be learning in an environment that's supportive and encouraging of taking small steps and understanding that those small steps make a big difference rather than, dwelling on the fear that you're not accomplishing everything all at once. So I've been thinking about what kind of environment kind of fosters this growth mindset, and how can that environment be fostered a little bit more? And as I think about that, I keep coming back to this paper from 2020 by Isabel Aubin that I cited in the review that I'm talking about today. So in this paper, she really highlighted the value of local organizing, of grassroots initiatives, and local communities of practice that support open data sharing within kind of small communities, whether that be academic subdisciplines or regional, geographic communities. And I keep kind of coming back to this as a really promising direction. And I think that meetings like this and communities of practice like Living Norway are super good examples of what this can kind of look like in practice. So I guess I just want to close my talk right now by just saying thank you to all of you for kind of engaging with this community and your communities, back at your home institutions, to continue building kind of incremental positive change with a growth mindset. And I definitely look forward to continuing to learn more with you, throughout the course of today and tomorrow about continuing to foster better biodiversity data management strategies together. Thanks for listening.
Now it's on. I think we have time for a few questions, Caitlin. But first of all, I would like to thank you for a really, really nice talk, and I think we had some really important reflections there at the end on how we can move this forward in a sort of welcoming way. I think that's what we really need to do. I have a couple of questions, and the first one is just reflecting on the questions you had in the title. Is the published literature a source or a sink? do you think? Is it contributing or is it...
I think loads of potential to be a source. I mean, I was surprised at all of the data that we're seeing that is of a presence-only data type, could be easily shared, that's being produced kind of outside of what we think of as the normal avenues for producing this data, citizen science, digitized data. So a lot more of this data is being created. There's a lot of potential there. But we're seeing that it's largely not yet really being fed into that open data infrastructure. A little bit, but not as much as it could be.
But in any case, there has been a really big growth in the amount of primary biodiversity data that are openly available. What do you think we can learn sort of when we are trying now to make the same happen for more complex ecological data?
Yeah, I'm glad that you asked that, because that was kind of something we talked about in the paper and didn't really get into this talk at all. Others this morning have talked about how this presence-only data is kind of the easiest to standardize. There are barriers, but it's less complicated. There's well-known best practices for doing this. So I think it's kind of like a bellwether, canary in the coal mine, or whatever expression you want to go with, that barriers that are preventing this type of data from being shared are going to be preventing all the more so more complicated types of data from being shared. So I think it's good to keep an eye on kind of trends in the sharing of this sort of simple best case scenario data, and use that to be proactive about issues that might crop up with more complicated data types.
Thank you. And if you want to continue the discussions with Caitlin, Slack is open and she will probably be there to answer and discuss. Thank you. Yeah, we are getting ready for the last talk before the lunch. And the last presenter will be Aud Halbritter from the University of Bergen. She will be presenting some work that is based on actually last year's Living Norway Colloquium, where we had some questionnaires and we assessed the participants' engagement with open science. I will help you. So when I'm finding your presentation, you can start to introduce yourself.
Thank you very much. So my name is Aud Halbritter, and I'm going to present you the results of a survey that we did with last year's. Thank you. So last year during this colloquium, we did. Okay. We did a survey with the participants, and we were wondering, or we were trying to figure out how open science practitioners apply and promote open science in ecology. As you have heard a little bit in the previous talks this morning, open science, reproducible and transparent practice are becoming more and more common in the scientific community. And for this to happen, a broad implementation of, I think this is, just needed to reload. I made some changes. Yeah, I think now it's fine. For this to happen, we need to implement, or a lot of cultural changes needs to happen across different layers, or different, like the individual, but also like at institutions needs to happen. And this includes, for example, different or new infrastructure. We need to redefine publication models. We need new data and metadata standards, maybe for some type of data like occurrence data. This is already more established, but for other data types like traits or ecosystem functions, this is maybe not the case yet. And then we also need some changes on like each individual on their research practice. And also, we've some of you in the previous talks have pointed out that these open science practices are not really widely implemented across the research community yet, but rather within smaller groups or selected networks like this Living Norway network. And also, there doesn't really exist one single definition of what open science is, but the different groups or networks, they perceive the values of open science and the skills that are required and benefits or trade-offs between costs and benefits very differently. And so a major cultural and behavioral shift among scientists is required to actually achieve the broader implementation of open science practices in our community. And we thought that a first step towards this goal would be to understand how open science practices and principles are learned, understood, and transmitted to other researchers and students. And we thought that this knowledge can help us to better inform or to better understand how open science practices can be implemented in the future. And this was basically our main question. How are open science practices and principles learned, understood, and transmitted to others? And specifically, we were interested in how, like a group of participants of such an event are defining open science, how they perceive open science. Then we were also interested in which aspects of open science they are interacting with and how frequently. We wanted to know what, like if you engage in open science, what are the perceived benefits and risks. And in terms of education, we were interested in which aspects of open science they have encountered in their own education. And we were also interested in, for participants that also are teaching, we're interested in how they value open science in terms of their own research, supervision of students, and teaching. We then designed an online survey. It consisted of 3 parts. We distributed the survey before the workshop, 1 part during the workshop, and then some of the questions again after the workshop. We collected some data, some background information about the participants, and then we had 2 different types of questions. Some were more quantitative to understand the experience with and perceived importance of open science in terms of research, teaching, and supervision. And then we had some more open-ended questions that were more targeted at understanding the definition of open science and what helps and hinders open science engagement. We then analyzed this data, these 2 different types of data separately, but then integrated them for the interpretation. And this is called a mixed model approach. And just to remind you that this is not the results of the study is not like something that represents the whole scientific community in ecology, but this was a survey done on like a selected group of ecologists that are interested enough in open science to come to attend the workshop like this. So just when we interpret the results, we need to keep that in mind. I think last year's colloquium had 128 participants, and 60 of those took the survey, the first part, 51 the second part, and then 39 the last part. Most of them were from Norwegian universities, but also other research institutes, and some were also attending from abroad. And then there was quite a good balance between women and men that took the survey. And this is the the result for the first question. So we asked the participants what their definition of open science is. And the most common answer was sharing data, accessibility or availability, and sharing codes and methods. And very, very few, I think only 3 participants, like right at the bottom, mentioned open science in terms of education. We asked this question before and after the workshop, and the answer didn't really change very much. But again, much fewer people took the second part of the survey, so we also have to be a bit careful with interpreting this result. This is one of the definitions that one participant gave and contains all sorts of open research protocols, methods, data, code, data management, analysis, and so on. And this is one of the answers that included open education in their definition. Then the second question we were interested in was in which aspects of open science the participants interact with and how frequently. And what we found is that the most frequent interaction is with reading open publications, using open code, and using open data. And this reflects very well like the definitions that the participants gave for open science. And then like sharing methods, code, and data, for example, is much less frequently used. We were then wondering how, for participants that engage in open science, how they perceive the risks of this engagement in open science. And it was interesting to listen to the talks before because some of these things that I'm presenting here have been mentioned. So I think they are maybe true. The most common things that risks that I mention is lack of guidelines, time, which has been mentioned before, and then insufficient knowledge. And again, this is a kind of representative answer from one of the participants, which mentioned lack of familiarity with relevant online platforms, softwares, methods, perception that the landscape of the above tools change very quickly, and keeping up is big time commitment and fear of doing it wrong. We also asked what the benefits are from engaging in open science. And here, again, this has been mentioned before, just in the talk before me, social support and resource availability. Those were the 2 most common answers. And one of the participants said abundance of open source, software, preprint servers, Science Hub, what a gem this is, GitHub abundance of data repositories, support from colleagues. To also understand how aspects of open science, kind of how the participants have encountered this in their own education, we were asking several different questions. The one here on the top shows that around half of the participants that took the survey, they have actually encountered something on using open code, open data, and reading open publications. But much fewer, like only a quarter of the participants, have also learned something about sharing your own code, your own data, or publishing papers openly. And then these things like open data, open code, and so on, reflect more the practical parts of open science. And then very few of the participants actually also had learned something more on like the ethical aspects or the principles of open science. So what does reproducibility or transparency means? And then the last question, we were curious about how they valued open science aspects in terms of their research, in terms of supervision of students, and in terms of teaching. And this was very interesting because we didn't really see any difference between research and supervision, but they valued these aspects lower in their teaching. And you can, it's not visible across all the aspects, but especially down here, when it comes to data sharing and code sharing, this is valued less important. And this kind of reflects this this paradox that we often see, that we see research and teaching as two different aspects. But yeah, that can be discussed. But I mean, they happen in the same place by the same people. So yeah. And so to sum up the results of this study, first of all, most of or generally open science was mainly understood in terms of shared and accessible data, code, and publications. And we saw that both in the definition of how the participants defined open science, but also in the aspects that they engage very frequently in. And secondly, they valued open science aspects very differently in their teaching compared to research or supervision. And by summing up like all the results from this study, we believe that by integrating open science principles and practices more formally into higher education, we can naturally address the implementation barriers that depend on individual experiences with open science. Yeah. So thank you very much for listening, and we submitted this manuscript very recently to PLOS ONE, so it's available as a preprint if you want to know more about it or read everything. And then all the codes for doing the analysis, but also writing the paper, is available on GitHub. And also this talk, by the way. Thank you.
So, first of all, thank you, Aud, for a very interesting talk, and I think it followed very nicely up from some of the points that Caitlin raised towards the end of her talk. A couple of questions before we go to lunch. Do you have any idea why these open science practices seems to be less sort of valued or used in teaching compared to science and supervision?
Yeah, that's a good question. I don't know. I think it's probably that open science is anyway something that a lot of us haven't really learned when we were educated. And it's also something rather new that we ourselves have to learn, so it's a little bit difficult to integrate that in our teaching, or to remember that it's important. If we want this cultural and behavioral shift to happen, then we should also teach it, because it's not so easy.
Another easy question. Do you think this is best done by having specific courses targeting open science, or that we should try to integrate it in more or less all courses where it's relevant?
Yeah, that's another good question. I think both could happen, but we have made the experience that we have some courses where, like field courses, where we teach students to collect certain ecological data, and there we try to start integrating different aspects of open science, like how do you make like your workflow more reproducible and stuff like that, and that sharing data and making it open available and documenting it is important, because this is kind of something that you can learn along the way when you collect ecological data. So I think integrating it to already existing courses where you teach other scientific skills is a good way to go.
Yes, thank you. And as with the other presenters, I think we can continue the discussion on Slack, or you can do it in person with Aud, those of you that are here at least. We are coming to an end of this first session. I personally have really enjoyed this session. I think there has been some really interesting presentations, and I encourage you to continue the discussions on Slack or in person here. On the program now, it says panel discussion. So the way we can do that is, if there are any questions in the audience here that you really want to address now and don't want to do it through Slack to any of the presenters, we can open for that. If not, discussions will go on through Slack, and you're of course free to discuss whatever you like during lunch. So are anyone having any pressing questions that needs to be addressed before? Yeah, Bob.
Thanks. It's something I think all of the speakers could comment on, is something Chloe said at the start about the sort of usability, having the tools that are usable. And to what extent is that being done? I mean, Living Norway, our package, is the obvious practical example, but others might want to comment as well. Is that, are we making the tools usable by your average ecologist?
Yeah, you want the presenters to... Yeah, it's tempting, since you mentioned Chloe's name. Maybe you want to say something, Chloe? Or is that making life too difficult for you?
Okay, well, thanks, Bob. Thanks, Alan, for the singling out. I'm tempted to say not as much as we should, and that's really where we have to do a lot more, I believe, because Living Norway R is a great practical example, and it's come a long way, and it's going to go much longer from where it is at the moment still, I think. But usability and user friendliness, I think, is really something we need to talk about also when we publish our code, for example, because the kind of the lack of standards and the lack of knowledge of the standard makes it so that it's everyone's own decision in what kind of form they publish their code. Like, I can't believe that I still encounter code pasted into Word documents these days, but it does happen. Or the fact that people publish the code for their model, but not the code for actually running their model. And I think overall we're not really doing that great. And I've tried to reuse other people's codes, and more often than not, it's a very frustrating experience. So I think we have to both define standards to make it reproducible, but what we really need to keep in mind is user friendliness. It's like a PhD student should be able to reuse the materials we publish, not just someone who has done the exactly same type of analysis for the last 20 years. So I think we have a lot to do.
Yes, I think you got a quite clear answer, Bob. Yeah, we have time for two more questions.
So my question would be, I think, addressed to Catherine Mandeville. And so she mentioned something that I didn't know, that 50% of records are from citizen science, and which is surprising to me. So my question is GBIF taking into account the possible errors, bias, which is coming from this fact that maybe citizens or volunteers are not so expert in recognizing somehow? Is there a way to take into account that the species recognition, for example, could not be correct?
Caitlin, you want to answer?
Yeah, I can say some about that. If anyone from GBIF is here, it would be cool to have that perspective as well. But I can say a little bit. So that figure about 50%, that came from the GBIF data blog in 2018. So I imagine the percentage is still high. I don't know if it's still 50%. So actually identifying citizen science data on GBIF is not a totally straightforward task. It's a little bit complicated. But that figure came from knowing that some of the major data sets on GBIF, like the single sources contributing the most data, are citizen science. So that's referring to eBird and iNaturalist and really large, well-known citizen science programs. So those definitely have a lot of quality control built in. There's other quality control built in, on GBIF where observations can be flagged with other potential issues that might be present in the data. The extent to which that happens, I guess, probably varies from data set to data set, but I guess a large chunk of that 50% is from pretty well-known and well-managed data, for sure. There's a lot more nuance there that I think I don't know enough to get into, but if anyone else here knows more, I'll pass the mic. Oh, yeah, of course.
I'm Dag Andresen from GBIF Norway. One very interesting aspect that we've been struggling with in GBIF to address this potentially is data annotations that you can go in and annotate with improving the data, or even just annotate that there is an issue. And to integrate that into the aggregated data made available is huge challenges, but I think that is maybe the most promising way to tackle this going ahead.
Thank you. I'm sorry for the online participants if this session doesn't work 100%, but we will have one more question and then we will go back to normal procedures after lunch.
Hi, I'm Joseph Driffield, so I'm one of the R package creators alongside Matt and Ireland. Actually, I was more going to revisit Bob's point about whether these tools exist and how far we are along with that. And obviously, the R package that we're creating is an attempt to try and bring some more users on board. But I think it is an important point to make, because I think from Al's results there, it seems very clear, and from Caitlin's work as well, it seems that a lot of the frustrations, I think, about people who want to engage with open science tools feel they don't have enough guidance and they don't have enough tools. And I think certainly I can talk from my own experience, is actually I think we can get quite far in ecological careers without even realizing what data standards there are. So it's quite hard to sort of know how to engage with standards if there's not really any education out there telling you what standards exist and also the tools to help you create those standards. So the R package might help some people along that way by providing the tools, but that still actually doesn't provide necessarily the education. I think so Darwin Core is an example of a standard, and I don't know how many of us in this room have engaged directly with Darwin Core or know about Darwin Core terms, but I would imagine it's not actually a huge percentage of us. And I imagine it's probably an even fewer percentage of us know about sort of some of the metadata standards, like the ecological metadata language. And so Matt talked a little bit about this being a particular headache for us. And I think this is still something that's very difficult to engage with the average user. I think so GBIF obviously started very well with this integrated publishing toolkit, which is a way for a lot of people to publish their data on GBIF as an example. But I'd still say that probably targets the more power user end of it, and I think that's fine if you have a large project and that you have the resources that you can hire people or have people in your institute like we do, and Nina here with Miliodata, who can help you refine your things for that. But it doesn't help for necessarily for these smaller projects, which might not really necessarily have the resources to do that. And I think for that we just need to create more tools in the hands of the researchers. Like I say, I don't know that the R package that we're creating will do all of that. I can't promise all of that. It still says it still is an R package at the end of the day, so we're still targeting users of R, which obviously there are plenty of researchers who don't use R. So we're certainly not going to get everyone, but I think that's definitely an area where we probably need to invest in a lot better to try and bring more people along.
Yes, and I think with that we will close the first session, and it's time for lunch. Next session will start at 1 p.m., so 1 p.m. Norwegian time. So I hope to see you all again then, and enjoy lunch.
Hallo, ja. Hallo. Så jeg setter den her nå. Hallo. Ja, for da forsvinner den nesten. Ser du det? Hallo. Jeg tror kanskje vi må stå der. Annette? Skal vi teste den nå eller etterpå, Lars? Ok. Ja. Hallo, hallo, hallo. Ja, det var det jeg tenkte på.
Ja, det var det du kunne, vet du.
Ja, men først. Ja. Så skal vi se om vi ikke har noe bedre å dele her. Nemlig.
Hallo, hallo, hallo. Ja, se der, det var... Ja, jeg har fått. Takk. Hallo, hallo, hallo. Var det sånn her det var sist? Hallo, hallo.
Nei, det høres jo helt fint ut.
Er det greit nå, er det ikke det? Ja. Ja. Hallo, 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 hallo. Lars, Lars, Lars. Det er greit nå. Ja, vi har jo stilt ned 13 eller 15 decibel der på mikrofonen, så... Ja.
Det var stilt for løft?
Nei, vi har... Nå funker den. Nå funker den.
bla 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 bla. Hallo, 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 hallo. Okay. Jo, det må jeg gjøre. Jeg trodde jeg var ute. Sånn. Ja. Og nå skal jeg ta share screen, og så preview. Hei, hei.
Det var lenge siden sist.
Akkurat i tide.
Ja, var det ene møtet til det andre?
Ja, det har vært helt heldig med timingen i år.
Nei, det er Open Access-uka i år, så det er jo vår travleste uke.
Ups, ja, men det var ikke noe sånt der. University of Connecticut. Det var litt sånn mangelfull liste over. Der vet jeg hvor jeg fra i hvert fall. Jeg måtte introdusere det. Jeg som skal skjæres i sol.
Ja. Det er stolen på sesjonen, det er bra.
Ja.
Kan du tenke deg å flytte opp det her i rommet?
Ja.
Og sitte her og så hjelpe å sjøfle på slides?
Ja, ja, jeg kan gjøre det. Er det noen som sitter der?
Nei, jeg har satt der.
Ja, fint. Da setter jeg meg bort der. For det er noe sånn, når vi skal ha den som du skal ha nå, så starter vi med Ingrid. Og så skal vi til USA. Eller hvor skal vi til USA, ja? Til han Daniel Bolnick. Så han er på... Sikkert ikke riktig uttale. Samme Dan, tenker jeg han er. Så da må vi slå av denne her, og så må vi ta inn et Teams-møte. Så vi må koble oss på et Teams-møte etterpå.
Ok, men det ordnet dere.
Jeg håper det.
Det er så lett panikkstemning.
Ja. Jeg tror det ordner seg, men det kan ta det går litt tid mellom at vi får liksom koble på da.
Jeg har slekket på slutten av sesjonen, så det er satt av en 10 minutter med søtte for å si kvartler til diskusjon etterpå. Så det er slekket det sånn 10 ganger til.
For nå...
Så liten krise.
Stop sharing, sant? Sånn. Og så skal jeg inn på kalender, og så join meeting. Og så går jeg ut igjen. Og så går jeg på share screen, og så preview på den. Ja, det skal ikke være vanskelig. Nei, jeg har ikke fått den ennå.
Hallo, hallo. Er den på nå? Høres det greit ut? Ja, bra.
Yeah, it's on. Thank you very much. So then I would like to have the pleasure of welcoming you to the session number 2 today. I will do my best to share this session. We will have 3 very interesting speakers. And the first out of them is Ingrid Hegeland. She's a senior research librarian at the Library of the Norwegian University of Science and Technology. So, Ingrid, stage is yours.
Thank you so much. So, first of all, I'm very happy to be here. It's always an honor to be able to attend this colloquium. It's a great place to be. So, I am from the University Library at NTNU, but I am not representing NTNU per se today. I'm here because I have been part of a national committee on sharing and licensing and reuse of research data. So, I want to present some of the highlights, what I think are the highlights from this report and our process and what we've been working on. So, for now, it's only Norwegian. It will be translated. It's on its way in English. So some of my stuff might be in Norwegian. It's a bit tricky to translate on the go, but I think you'll get the gist of it. So I'm talking about some highlights because the final report is like 70 pages or something, so you can read more details if you're interested on your own. So some background. Last year, early summer, the government said we need to figure out this stuff about licensing and ownership related to research data. So they said we want to share and reuse research data. We need a committee to think about the legalities and how to organize licensing and so on. And this is the committee. We have Magnus Alesen from EU. He was a leader of the committee, and we are a group of different people from different parts of research, science, innovation in Norway, and different fields of research or fields of competency. And we have had great support from the Research Council. They were our secretariat, and they helped organize and help write and really influential in the process itself. So we started out, we got this mandate. It's in Norwegian, as I said, but the point is looking into rights and licensing related to sharing of research data, looking at the legal issues, legislations related to this, and what are the consequences when choosing licenses for research data. And the goal is sharing and reuse of research data. So this was the overarching mandate. So we started in June 2020, during the pandemic. All our work was during the pandemic, so we had Teams meetings. I met this group once physically. It was very nice. But everything virtually. And we quickly realized that sharing and reuse and licensing of research data, it's more than just the legalities. So we started with a memo that we had ready in October 2020, kind of drawing the big picture and saying, here are some issues we'd like to discuss further before we have a final report. And then we had a period of open consultation. We got feedback on the memo. We had meetings, we had webinars, we had conferences, trying to engage the different stakeholders in this process. So I hope some of you noticed that we had a process and attended some of these things or read our memo. And then we wrote a draft for our final report, which we sent out in June, just before the holidays. Lovely. And we got a new final round of feedback from the stakeholders. And now, just last week, we released our final report delivered to the Ministry of Education and Research. And then it's up to them how to follow up. But now the report is out. It's there for everyone to read. And hopefully soon also in English for everyone to read. So I want to highlight a few things that we discussed in the memo and also in our final report related to research data sharing and reuse. So we also emphasize all throughout the report, research data is a lot of different things. So this is the EOSC definition. So all data collected or produced in the course of scientific research activities and used in evidence, this is what we talk about as research data. And I know that in some fields, maybe not your fields, they say, well, we don't have data, we have sources, or we have used different things, they're not data per se. We include everything you use to make an argument as potential data. Also note that we often talk about digital data, but not specifically, but in practice we often mean the digital data or data that can be digitalized and shared in a digital world. I'm not excluding the physical data, but this is how we can actually share it. So the point is, everything that can be considered research data we include in this definition. And we talk about sharing and reusing, and we also want to emphasize, we of course want open data, but even more importantly, we want the fair data. We want the data that is described and organized and sensible and reusable, even if it can't be fully open. Still, it should be described well and it should be understandable. And I think this, looking at the descriptions of your talks, you all know what fair data is, so I won't go into details. So a large part of our report and our work was actually looking at the legal framework. And to summarize the legal framework for research data in one sentence, it's messy. It's all over the place. We don't have one research data act or law. So you have to navigate many different laws and regulations to really figure out what's relevant for you and for your context and your project. But that doesn't mean it's always very difficult in practice. It just means there's a lot of things to consider. We have the really overarching principles and acts like the Open Data Directive from the European Union. We have to consider that. Many of our national laws are, of course, in alignment with the European legislation, thankfully. For me, at least, I work at the university. It's a public institution. We have to consider the Public Administration Act, for example, and also the act relating to the university and university colleges. For all people doing research, the Act on Ethics and Integrity in Research is really important to know about. And the Health Research Act is one more specific act. So many of these, they have some either clear requirements, or they state some boundaries or expectations related to research data, research output, how it should be managed, handled, and what should be public. So to summarize these in general say research data from public research should be openly available if possible. And then we have some legal framework that might work in the opposite direction, that might say this data cannot or should not be shared, at least not openly to everyone. Examples are the GDPR and the Security Act, things that would harm people or maybe harm the national security, that should not be shared openly. Also the Copyright Act, that might say that you have certain rights as a single person or research or institution, and you can say that means that I want to restrict who has access and in what form and what they can do with it. And in some cases, laws for specific fields of research. In your case, maybe the Nature Diversity Act is something you should consider when you're thinking about sharing and publishing your research data. Some cases you might say you have to, in some other cases maybe you can't publish openly, for example. But maybe the most central one when deciding if you're at the point this data should be shared openly, how do I license it? It's the Copyright Act, in Norwegian Åndsverkloven. So who decides what license to put on? What are the rights regulating research data? And again, it's a bit messy, but a lot of research data is not actually covered by the Copyright Act. Facts about the world, they can't be copyrighted. So if I measure the temperature in that room, that's not a copyrightable fact. That's a fact. And note that the Copyright Act, the thought behind it was artistic expressions, åndsverk in Norwegian. So research data is sometimes a bit slightly a gray area, whether it's covered or not. I won't go into the details, but what we have done, we've made some lisensrettregler in Norwegian, a license code, if you may, so some principles when you think about how to license and share data. Note this is at the point where you say this data can be shared openly. There's no restrictions based on it, like personal data and so on. Some aspects to consider. And to summarize shortly, if you publish or share research data, put a license on it, or something describing what are the requirements when you reuse it. Other things, as open as possible. Don't put restrictions unless you have a good reason, like adding a non-commercial license. Really think about, is this necessary? What is my motivation to do that? And also, if you have standard licenses, it's so much easier for the rest of the world to understand what they can and can't do with the data. And interoperability is also important. So when these are translated, a bit easier to understand for those of you that don't speak Norwegian. And the last point, very important point, whatever license or public domain dedication is used, when data is reused, you should always cite and quote what you are actually using in regards to research ethics. So as a researcher, you should also always say, this is where I got my data from, this is where I got this information from. So that was kind of the legalities that we have discussed in our report. And then we also say, well, that's maybe not the hardest thing. How do we actually do this in practice? How do we make sure that we actually publish and share research data and reuse? So we have some recommendations that are maybe a bit outside our mandate, but that we think are important to address. And they are at the level of the government. We have some recommendations for the funders, like the Research Council, the Horizon Europe, and so on, and some for research institutions and also the fields or the actual researchers themselves. So I just want to highlight a few of the things or topics we recommend. So to be able to share research data in a sensible way, we have to have policy alignment. So we encourage institutions, the government, to look both nationally and internationally, what are sensible guidelines, what are good practices, and make sure it's possible for researchers and research groups to do things in a sensible, useful way. We also think to actually get something done, we need some incentives. So in Norway, we have the so-called Tellekant-systemet, and if you don't know what it is, good for you. Because I think we should have a different system. That's my personal opinion. But the funders, the institutions and the government, they can influence how do we publish, what do we publish. Do we also include research data as a type of publication, for example? The funders could say, well, we want to look at open science practices when we give projects funding, and so on. They have started doing some, but we still have a long way to go. Also, the institutions themselves, when they hire people, if you are applying for a professorship, maybe you should show how did I share data, how did I share my code, and so on, more than just your impact factor for your papers. Also, how do we do it in practice? We need good infrastructures, we need good repositories and archives, and we need competent people that also train our new PhD students, our master students, and of course our senior researchers. Note the order I did this in. So this might imply a culture change, not for all fields of research and not for everyone, but as a whole we have to change the way we do and the way we value science and science output. It takes resources and time. We have started, of course, but we need to really prioritize this for this to get rolling. And finally, we talk a bit about innovation and commercial potential because we also want to make sure we collaborate with commercial potential with outside the universities and the private research institutions. So think about who can reuse the data. What do we have to do to make the data interesting outside the research institutions themselves? So not a small task, but we are at least moving forward, I think. So thank you very much for your attention.
So thank you very much. This opts for a lot of interesting discussions, I think. We have time for a few of those moments now. So first of all, I'd like to take something from the Slack feed here. So the question is, did you consider also the Environmental Protection Act or Miljøinformasjonsloven in your report? So because this might be quite relevant for a few of the data types we are considering within our network.
Yeah, so not specifically, because there's probably a lot of specific laws for specific fields of research that really have to be considered in that context. So but the considerations would be similar. You look at the law and see, are there any specific restrictions or specific reasons to share or not to share? So that's the easy answer. And these considerations often come before the point that you decide of the license. So all these complicated things we haven't discussed in detail because that would be, really a lot. But it's an important thing to consider.
Yeah, thank you. Anyone else from the audience? Yes, Chloe. You have to talk into the mic, I'm sorry.
Okay. So that was really interesting and a very different angle on things. You mentioned that it's really important that there's some consistency between the regulations and recommendations at like different levels, and you had there both nationally and internationally. So I was wondering if you know a little bit about how the rules in Norway relate to the rules of the European Union or the rest of the world. Like, do we have a chance of having something consistent for everybody and not just for Norway?
That's a very good question, and I don't have a simple answer. And I don't have legal competencies apart from being interested. We are lucky to be in the European Union, I think, because many of our legal frameworks are at least harmonized to a certain level. And we also look to the European Commission in regards to research and what they think are important. So I think in practice often it's possible to do things in the right way and not be in disagreement. But I know maybe in particular working with the US, for example, it's not always easy to harmonize all details and all practicalities. And also we have some of the practical issues with copyright law, not the same, especially not in Europe and the US and so on. And sometimes we have practices that have evolved but haven't really considered all the legalities, like the use of Creative Commons licenses on research data. It's maybe not optimal, but at this point maybe we just have to go with it because that's what's well known. It's not a complete answer, but it's a very large and complicated and interesting question.
Yes, thank you. Very brief question in the Slack here. Did you consider DORA or for also it might be aligned with that?
Yes, I can't remember if he specifically mentioned it or not, but absolutely we discussed DORA. DORA is a great initiative. We were just a bit careful about recommending very specific things towards the end. But I think we mentioned DORA in the report somewhere. We have certainly discussed it many times.
So DORA is the Declaration of Research Assessment, by the way. So for those who don't know that, we often get criticized in this community that we are about a bit too fond of acronyms. Yeah, this was a typical example. This is also in a question in Slack from Erlend Nilsen. Do you want to take that in person, Erlend? Or with the mic? do you dare to?
Thank you, Ingrid, for a great presentation. Well, my question was more out of interest. So you mentioned that you went beyond your mandate, and I personally am glad to that. I like the report. But I was just wondering if you got any objections or comments or things like that from those that initiated it, or was it all fine?
It seemed all fine. We had a very interesting discussion in this process, and to be honest, I don't know if they noticed that we went outside our mandate to start with, because it kind of evolved. But I just think it's very important to say that licensing and legalities related to research data, yes, important, but it's just a very tiny bit of what actually is needed to make sure we really share data. Yeah. So very quickly we said, well, we can't just look at the legalities. That would make sense.
There we are. Thank you. If there are not any more questions specifically, there are, yeah, one. We have very, very brief one. Okay.
Yeah, maybe mine is more a comment rather than a question. I was also thinking about the presentation from Chloe this morning, and yes, so some of the things you mentioned, Ingrid, about the importance of the incentives about so and the comment about the Chloe presentation is one of the main blockages in sharing considered to be the lack of time. But from my perspective, and I'm not a researcher, so probably I have a different perspective, but the lack of time, the perception of lack of time, is relative to the importance you give to the act of sharing. And this is also related to the incentives and how this is being considered important in the scientific community. So, so we are facing probably a shift in which this aspect compared to publishing articles and journals and so on was more important in past, and now also the data sharing is becoming important, but you will find the time if you consider it important.
Thank
in
you.
My opinion.
Yeah, I totally agree. I hope it will be like saying, well, I don't have time to do my research. When you say I don't have time to publish my data, make it fair. You don't say I don't have time to write the paper, right? It's the same. So it's just a shift in time, I think.
Thank you very much. We have to move on to the next speaker. So applause again. So now we are moving abroad. And next is by...
Okay, so then you're on.
Then you're on. So I'm very happy to introduce Daniel Bolnick, which is editor-in-chief of the American Naturalist Journal, and is also a professor at the Department of Ecology and Evolutionary Biology at the University of Connecticut. Welcome.
Thank you very much.
So then I leave the floor to you.
All right. I'm assuming everyone can see and hear me okay. If not, let me know.
It's all very good.
Excellent. I appreciate the chance to talk to you all today. And what I'm going to do is give just a quick overview of data sharing as it's pertained particularly to the journal that I'm involved in. And the issues that we see at the American Naturalist are quite, I think, generally applicable. And so I think it's a good case study, hopefully. Just for some historical context, the journal that I edit, the American Naturalist, is a little over 150 years old. And in its early days, it was largely data-free. It was mostly natural history observation, but with nothing numerical or quantitative. Really quantitative analyses began to emerge much more in the late 1800s. And by the early 1900s, especially the advent of genetics, really turned the discipline into a much more quantitative field than it had been previously, with the introduction of a lot of statistical analysis emerging as well. And often the data was actually inherent to the paper itself. So just as an example, this analysis of sex determination in humans, published in 1907, the raw data was actually printed as a table in the paper itself. And this was quite commonly done. And the data files were often small enough that the raw data could be simply provided in the paper. But of course, as our data sets began to grow, they outgrew the ability to just put the raw data in as printable tables. And so that tradition really ceased, and we wound up in the situation that we found ourselves in through the past decade. The very first online publication of the American Naturalist was in 1999, and then the first appendix of raw data was published in 2000, 20 years ago in the journal, 21 years ago. And so that was a step forward, but it was not consistently done. It was simply at the decision of the authors. And the problem, of course, is that authors would typically write something like, data is available upon request. And they were expected to honor such requests, but everyone knew, wink, wink, that it didn't actually happen. And just as an example, I started to do a meta-analysis a few years ago that required not the published summary statistics, but the raw data to apply a new statistical approach to estimates of stabilizing selection. And so Joel Kingsolver had done a meta-analysis on disruptive and stabilizing selection around 2001. And I emailed all of the authors of those original papers that he was using and asked, could I get the original data? And you can guess how many of them actually supplied that original data. The answer was about 5 out of 120 people who I emailed, which was terrible and very frustrating for me. We never did the analysis that I hoped to do because we simply couldn't get our hands on the raw data. And there was a paper in PLOS One in 2009 that highlighted the issue. They did 10 requests for original data. And those 10 requests, 2 of the 10, the email didn't work anymore, so they simply couldn't contact the authors. And 8 did have functional email addresses. 4 flat out refused. 3 didn't respond, and only 1 was willing to share the data. And so of 10 requests, they only received 1 successful outcome. And authors cite a variety of reasons. Too much work is one. I no longer have the data file. It was on a computer, and that computer is dead, or the computer was stolen. It's surprising, actually, how often the laptop was stolen explanation gets cited in some very prominent cases. And so the conclusion has been that available upon request is not good enough. I think that's been clear for well over a decade, and it surprised me, actually, in preparing for this lecture, to see this paper from only 3 years ago saying available upon request isn't good enough. I would have thought that this conclusion had been quite clear long before 2018. Now, the tradition of having documentation of our data goes back very far. Of course, museum specimens in biodiversity research and taxonomy have long been the standard. If you collect an organism to describe a new species, you need a type specimen. And if you don't have a type specimen, you don't have evidence of that species. And so museums have long served as a repository for data of a certain sort. Then the growth of GenBank and DNA sequencing technology meant that we saw very, very fast accumulation of online data archives of DNA sequence data. When I was an undergraduate and was taking molecular genetics in 2004, 2005, I was sorry, not 2004, 1994, 1995, I was already able to go on GenBank and blast a sequence and find matches. And that repository feature was crucial to DNA progress in a lot of genetics and was very standard, and nobody raised particular concerns. Inspired by that tradition of both museums and GenBank, there was a group that met at the National Evolutionary Synthesis Center in 2007, led by Todd Vision, among others. And Todd Vision had a vision which was requiring data archiving, and that led to this paper by the editors of several journals, including Mike Whitlock and Mark McPeak, who were current and incoming editors of the American Naturalist at the time. In February 2010, they published a paper arguing for requiring data archiving, and that requirement became live at the beginning of 2011. And the benefits they argue is that it archives the data safely so that, for example, if I post my raw data and then my computer is stolen or my computer dies, my data is available for me on a public repository that's outside of my own control, which protects it from my own mistakes. It makes it available for reanalysis and meta-analysis that go beyond the summary statistics that I might have chosen to include in my publication. And, of course, data can be checked for errors and for fraud more easily if the raw data is available. And we'll return to that issue in a few minutes. There was an acknowledgement that there were costs to this. There's a sense of ownership that authors have, that it's my data. And often that sense of ownership goes along with the intent to publish more papers out of that existing data set. Sometimes that intent is realized, and often authors might plan to publish more on that topic with that data, but in fact, they never get around to it. And so there was genuine and valid concern that that by publishing raw data, authors might be scooped in their own ability to publish their own work subsequently. And then there was also a concern that secondary users who might visit and use my data to publish their own papers, maybe they don't acknowledge me, maybe I'm not a co-author, but also maybe they make mistakes because they don't understand the biology of the organism, they don't understand the specifics of the experimental design. Data by itself can be subject to misinterpretation as well as misuse. Now, in practice, many of these concerns, I think, are very overblown, but they did have a significant impact. The initial response by the community was not uniformly positive. I remember conversations at the European Evolution meeting in Uppsala in Sweden in 2000, I think, 10, when this was first coming down the pipeline. Maybe it's 2009. And I remember having conversations with people who were really quite upset by the prospect of this requirement. And submissions to the leading journals that adopted this policy in 2010, 2011 really did drop. The American naturalists saw a real decline in the number of submissions at the time that this policy was instituted. And some other journals like Ecology that did not have that policy and actually didn't until very recently went up. And so we did lose market share in a sense as a consequence of that leadership. However, a lot of the concerns seem more like laziness than anything else, because today we do have a policy where authors can request an embargo, a waiver, in certain situations. So for example, if the data includes information about individual people and the data is collected with a requirement from regulatory agencies that the information be anonymous, then elements of the data have to legally be redacted, and we're able to accommodate that. There are also embargoes where we say, well, we'll put the data on a repository so it's safe from author manipulation, and we know it will be available, but we can put a longer timeline on access by viewers, so that there's a delay, and that delay gives the authors more time to publish more papers. And so that we can be somewhat accommodating within reason. But I have, in 4 years as editor, received 0 requests for waivers or embargoes that had any reason behind them. I've only received about 3 or 4 requests for any kind of waiver. And in every case, it was simply, we don't want to bother. And if I pushed back, the author said, well, okay, I can do it. So the number of packages that are actually archived on Dryad did in fact increase very quickly and continue to increase very quickly. And so it's been very satisfying to see this policy successfully implemented. And as this changed, we really have seen increased acceptance of this material. And it has proven itself to be absolutely crucial in retrospect. I can't imagine having been in the situation of not having had this requirement. So most famously, a particular researcher who studies spider behavior has had a series of papers retracted at our journal and at other journals as well on the basis of problems with his data. And this was a situation where this particular researcher was often a co-author and would have conversations with colleagues about an idea and then would email them a data file 2 or 3 months later saying, hey, that idea we talked about, well, I did that experiment. Here's the data. Do you want to write it? And what we discovered, I'm sorry, these screenshots are a little blurry, is that there were duplicated batches of data. So for example, this block here highlighted in yellow has a 0.0073, 0.0068, 0.0091, and the exact same numbers are duplicated in the exact same order down below, ostensibly on completely different spiders. Same thing with these data blocks highlighted in gray. We have, again, exact duplicates with some intervening data that is not duplicated. So there was clearly some kind of error or manipulation. There are instances as well by the same author where there are Excel data files posted on Dryad, and when you look at it, it looks fine, but then you click on a cell and it turns out that there's a formula there, and this change in mass is supposed to be the dependent variable. He is regressing column D on some of these other variables. In particular, is D a function of E? Well, it is because there's a formula that calculates D from E. So supposedly the dependent variable in his statistics is actually being generated through an algorithm rather than through data collection. These cases have resulted in retractions, obvious either mistakes or fraud, and it looks very much like fraud. From a legal standpoint, I'm being very cautious in my phrasing. There are other cases that are even more egregious by the same author, where these blocks here in green and in yellow are shared between two papers that are actually ostensibly from different species of spiders in different locations in the world on different years, and yet they're blocks of identical duplicated data. Clearly problematic, clearly a basis of fraud, only detectable because the raw data was required. And I was just speaking the other day with the editor from Ecology, and at the time that this author was publishing in the journal Ecology, they did not have a data repository requirement. And so they have a much harder time coming to a decision about what to do with this author's papers because they don't have access to the original data files to evaluate what was going on. Now, it's not to say that we are completely in the clear either, because, for example, in the case of another author, concerns were raised about some of the experiments and data by another author in behavioral ecology, but a lot of the data files were missing and incomplete. So although there were data repositories, they didn't contain everything that they needed to, and the author was unable to furnish the complete data set because he claimed that his laptop was stolen. And I was able to confirm that there were a series of car burglaries that included a stolen laptop, and that this individual was indeed a victim, as confirmed by legal authorities. Now, whether the data were never backed up anywhere else is an issue. So what we've done now is we've begun checking for data compliance. So we would always check before publication to ensure that a repository existed, but we never went into the fine details because it's a lot of work to check every single thing the authors say they have against whether it's actually a column in their data file, and is the metadata good, is the code readable. And so we've started doing this. We have a data editor, Bob Montgomery, with assistant data editors. And so far, we are 19 papers into this process. Only 5 of those 19 have very high quality data repositories with raw data, complete, metadata is clear, and code is available. 6 others have modest problems, some important element missing, but it's easily fixed. And 8 were really dramatically low quality. They were not usable to reproduce the outcomes of the papers. So although everybody is creating repositories, those repositories are not all equally high quality. Bob Montgomery also reevaluated 100 papers published in 2020. Of those 57 used data, all of them had repositories, but only 7 out of those 57 were complete and absolutely usable from the code on up. And so we're really trying to now enforce good data practices, not just is there a repository, but is it complete? Is it clear? Does it have every variable? And in doing this, we're partnering with a company called DataSeer. We had a trial run with DataSeer that uses AI to evaluate papers and come up with a checklist of what needs to be included. And that's turning out to be very helpful. And so Dryad was established in 2009. We require it, data archives in 2011, and in 2021, editorial manager is now integrated with Dryad, so while submitting your manuscript, you can also submit your data archive at the same time, and simultaneously we have data editors evaluating each accepting manuscript to ensure compliance with our standards. And code archiving is about to be required as well. We've highly recommended it, we've encouraged it, but we haven't required it, and we are about to do so whenever code is used. And so the workflow now is that authors submit a manuscript and simultaneously they're uploading data and code to Dryad. The journal pays for the Dryad repository, so it is free to authors or covered in their publication costs. the reviewers can see the code and data editors can see the code and data as well. So it's visible, but it's only made public upon publication. And we're using, as I said, Data Seer to help the data editors. And there are a lot of open questions still that we need to deal with. How do we standardize for diverse kinds of data? What do we do with very large data files like videos or photos or DNA sequence data that can take terabytes worth of data? And how permanent are these repositories? And so with that, thanks for your attention, and I'm happy to take questions in the little time that I left for myself here.
Thank you very much. This was interesting, slightly worrying, but most of all, I think it was engaging. So I think we have time for, we have precious little time. We are running out of time, but we could take a few quick questions or comments. I do want to take one from Slack, actually. This is Rukaya Juhadin. She is not sitting here in Trondheim, so she's out there in Oslo, and she is commenting that she's a bit surprised that the peer review process not always does a thorough job of checking code and data. Could you brief comments or thought about that?
Well, it's a significant extra, I mean, I agree, but it's a big demand to place on reviewers who are volunteers. Not all reviewers are conversant in the code that a particular author chooses to use. I just had a submission that used JavaScript, no, Fortran for the code, and I don't speak Fortran, so it's hard to, because code isn't standardized, the languages aren't standardized. It's not possible to demand it always, but it's something that we're moving towards, but it's an additional burden on reviewers. And I think there's been a lot of fear that it's too much to ask, especially sometimes code takes hours or days to run on fancy computer clusters. It's not always possible to check through, given limited time and computing capabilities.
Thank you so much for that answer. I'm afraid that we have to move on into the program. We're a couple of minutes on overtime already. If you have time, you could hang on. We will hopefully have a 10-minute session at the end of from 1300, 10 to 4. This will be Norwegian times only, but hopefully we'll get you in for the questions afterwards. I'll talk to the tech guys about. In the meantime, I'm extremely happy that we can now present Colloquium. He's a data management coordinator at Elixir Norway and at Biomed Data. So please, and your presentation will get up in a few minutes. Yes, thank you. You could use this microphone, or do you want to use any?
Okay. Thank you very much for this kind introduction, and thank you for the opportunity to speak here. I would like to pick up where Dan left us on the sequencing databases that he mentioned, and would like to touch a bit upon this and how they link for ecology data. So I'm affiliated with Elixir Norway, Biomed Data. This is an initiative on a European level on sequencing data and on other molecular life science data, and we are coming from this angle. So, of course, I was inspired on this because I read this report from Anders on the flow of data in ecology, and then I had a look at this, what is squeezed here on the edges on the sequencing databases and on the metadata standards for sequencing data, and I thought it would be a good opportunity to introduce this a bit more. So the network of sequencing databases has also a geographical part. So if we look at the different sequencing databases, there is one from NCBI in the States. There is one in Europe, which is an Elixir core data resource, the European Nucleotide Archive. managed by EMBL, and it's also used by Argentina, Israel, and Australia. And then there is the Japanese Gene Bank. And they are actually linked to each other, so they exchange data on a continuous basis. So regardless from where you deposit your data, it will be available through the other databases as well. And they just turned 30 years this year, I just learned. Unfortunately, in addition to that, there is databases that use the raw data, so the sequencing data that are sitting in these databases, and they have additional data like additional metadata or process data, and they sit on top of these databases, and they are not necessarily all of them synchronized, and they don't exchange data. So we have, for instance, Geo, that is a platform for expression data of RNA-seq data, for instance, and we have ArrayExpress in the States. And earlier there was an import from GeoData to ArrayExpress, but not the other way around. And this also has stopped in 2017 now, and it's only done for key data sets now, but only in the direction from Geo to ArrayExpress and not the other way around. We come back to these value-added databases a bit later here. So which repository is the correct repository for your data? We have different types of data here. So we have next generation and third generation sequencing technologies from Illumina sequencers, for instance, that you might use. Then we have capillary reads, so like, for instance, Sanger sequencing that you might use for barcoding. We have annotated sequences, and we have studies and samples. And this is a little complicated. So for the Japanese data bank, we have one different repository each, but luckily in the case of ENA there's only one resource where you can go regardless of the data that you have. And then again for NCBI this is a little more complicated. And you see here the bottom line, and we come back to this again. There is biosamples in each of these three databases, and in the case you generate an entry in ENA, this biosamples entry is also generated automatically for you, so you don't have to do that separately. And that's also where the interesting, probably, information for you sits. So if you want to register data in ENA, this is rather straightforward. There's different possibilities for that. You can use the interactive submission, using a web interface. There is a command line tool, and then there is also the possibility to use an API to submit your raw data and your metadata to ENA, for instance. And in any of these cases, you also can use the embargo function of ENA that is rather generous. So you can put your data by default under 2 years of embargo, and you can then lift the embargo when you publish the article, or you can extend it afterwards. That's also the reason why many sequencing consortia in the States are using ENA and not NCBI. So if we look at what an entry in one of the INSDC databases actually entails, we have 4 or 5 different parts here. We have the study itself, that is the whole submission, and this includes the information about the experiment and the authors, for instance. Then we have the run, that is the raw sequencing data. We have the experiment, that is how the sequencing data was generated, and we have the sample. So this is in the study we have the involved persons, where they are affiliated, where this is based. Then we have in the experiment wet lab steps, what the sequencer, how the sequencing was done, and how the what flow cell, for instance, you used for your sequencing. And then we have the sample, and I think this is probably the most interesting part for many of you, because that is where the information about the individual or the taxon also sits, and there is a data standard, the so-called MixS data standard. So looking a bit at MixS, this is actually consisting of 2 parts. So we have one part that is related to the technology. So what do we sequence actually? So there's one for genomes, one for metagenomes. If you have a lot of different species in one sample, and there is one for, oh, this is wrong. Yeah, that's twice the same. Okay, there should be one for amplified sequences as well. And then we have a second part here on the environmental package. And that is different depending where the sample was generated. I left out some human-associated packages here, but you can see, like, depending where the sample was taken, you have a different checklist, and they're, of course, also overlapping. And then these lists are populated with different controlled vocabularies that are either inbuilt in the standard, or they are using an external ontology to represent, for instance, the species using the NCBI taxonomy, or they are using the EFO ontology to describe the experimental steps that you have taken to generate the sample. Okay, how does it link with all the other databases? So we have ENA here in the middle, and then we have other databases around. So for instance, we have MGenif. You might have heard that. That is a database for metagenomic samples. So if you send a sample to ENA for metagenomics and you flag it as a metagenomic sample, MGenif will pick it up, and they will do a standard processing pipeline to ensure that all data in this database are analyzed using the same workflow and that they are comparable. And then we're coming back to how they link to GBIF later. Then we have other databases like RNA Central or UniProt that use data from ENA. And we have, for instance, Array Express that is used for expression data and contains additional metadata. And then using a similar process, doing standard analyses to Expression Atlas, so that you can use data, directly process data that are analyzed using the same workflow, so that they're standardized also in their processing. And just as a comment, if you're doing qPCR data on really large panels for instance, for environmental DNA, you can also deposit them in Array Express. And for the big reference genomes ensemble, the raw data is also stored in ENA. Okay, how do GBIF and ENA link? There is different links indeed. So we already talked about MGenif, so they are directly actively generating Darwin Core and make this publicly available in GBIF. That is one possibility. Other possibility, I think this is very exciting actually. There is now a crawler that is called the EMBL adapter together with the GBIF clustering pipelines, and that crawls ENA and generates Darwin Core entries from this and pushes them to GBIF. And then you also have the possibility, if you already have created an entry in GBIF, that you link to an ENA entry manually or semi-manually, or just as a dedicated link. So you can enter in the Darwin Core fields associated sequences and occurrence ID, the entry that you have in the INSDC, or you could also use the MixS extension of GBIF to populate this with the information that we talked earlier about. If you're using Bolt for barcoding, then this is not linked directly with ENA, it's only indirectly linked with ENA, but you can, they are partially importing data from GenBank, but you can also, there is a function in Bolt to directly submit data to GenBank, but you have to trigger that on your own. Okay, I also would like to highlight here that we have a tool to help you with planning your studies. It's called the Data Stewardship Wizard. And thanks to Rukaya, who is joining remotely today, we also have recommendations for biodiversity in the Data Stewardship Wizard that are suggesting some data standards, for instance. And we also have suggestions for any kind of non-human sequencing data that you can find directly there in your data management plan, and you can complete it on your own. We also have pipelines and infrastructure to do DNA and RNA analysis. If you would like to use them, it's called NELS, Norwegian Infrastructure for Life Sciences, and there we have reproducible pipelines in Galaxy that you're welcome to use. And we have a support help desk also for that, and this is free to use for research projects based in Norway, enabled by funding from the Research Council. Okay, I would like to touch up on a second topic also. That also came up when I was last year here, because over dinner we discussed about sensitive data and sensitive human data, and then I thought this is not super different to sensitive biodiversity data. There might be some stronger legal requirements and to it, but then in the end it's about like protecting the access to data. And I looked a bit into this because we're also involved in the Earth Biogenome project now, and I read these guidelines from Milieuddirektoratet, and they have 3 different categories for sensitive data. So they have some that can, or 2 sensitive data categories. So they have some that can be open and have to be masked, and they have you probably know that better than me. So they have this password protected, and they have this restricted access categories. And then I realized that this is very different for different national portals. Like if you're looking at NBN Atlas, they do that on a feature level. So they have 10 different criteria, and then they grant access to data providers depending whether they need access to different features or not. And in GBIF itself, this is put into 5 different categories and is rather complicated. And they don't necessarily map one-to-one. And what that reminded me of then is the system that we use for sensitive human data, where we have an ontology to describe exactly all these different access categories. So there is the data use ontology from the Global Alliance for Genomic and Health. And this is describing, then you can say, for this type of study, this access can be granted, or for this type of, in this geographical restriction, if you're based, say, in this Fylke, then you can get access to this wolf data, or you can get access depending of your affiliation also. So this is a it's a very fine granular way to express who should have access to a data set, and it's also practical used in the European Genome Phenome Archive, where we have controlled access to human sensitive data for instance, for health research. And then you have to do a request to a data access committee, and they judge based on this machine-readable ontology in order to give you access or not. So I thought that maybe that's something worth to look into also for sensitive biodiversity data, to have a higher standardization of the access terms and to be able to map the complexity not only in rough categories but also more fine granular, depending how the data should be accessed or not. Okay, I hope I'm still on time here, and would like to thank again. And we are funded from the Research Council of Norway and the universities and the European Commission.
So, thank you very much. I was hoping that we could have time for one more specific question for you before we move on to the more general discussions after this session. So, I have to open my Slack, or if anyone in the audience probably want to have something. So we will continue with some general questions to the session. We hope we can take it to there. There's a lot of questions on the Slack forum here. We don't seem to have any connection with the US, do we? Or is it? Okay. I think we'll just start, and I can see this. We'll take some brief questions in between with to see for Colloquium here, where Dag, you have a question regarding data quality or standards for genetic data.
But it might be a bit too nerdy, but you link through the occurrence ID, which is a species in a location, and the mix could maybe stronger linked through the material sample ID in the Darwin core. So I guess it was just, yeah, it was very nerdy, but a small detail.
Okay, there we are. Thank you. So, there's a hard to pick all these good comments and questions, but I think one that is reiterating here is regarding reproducible code and on how to ensure that published code actually is reproducible. So anyone of you like to comment that? I guess that goes most of you to Dan, though. Is that you? Do you want to answer?
A particular question, sorry?
The question has been on how to ensure that actually, okay, you will say that your code should be public, available along with your paper, but how to actually ensure that this is reproducible and running. What are your thoughts about that in the future?
That's a very difficult problem, and it's one of the things that has kept us from implementing code requirements as much as I've been wanting to. I commented already, some code takes days to run, or weeks, and requires a supercomputer and can be very expensive to run. And I think we're just never going to be, in my own lab, we just completed an analysis that took 2 weeks to run and cost us quite a bit of money on the supercomputer. We can never require that a reviewer has access to those computing resources and the financial backing to run it. So that's an extreme case, of course, but when possible, our data editors do try and run code, and they have been, in some cases, able to flag problems in code. I think the main thing right now is our minimum is it annotated well enough that the editors know what a piece of code is meant to do, looking at the different pieces and say, okay, this is this analysis, this is that analysis. So we're asking for improvements at that level, but we're just not always able to run it. And it, of course, depends on having the right version and the right package. And there are ways to create archives that are a version of your particular version of R with your particular package versions, but not everybody is savvy enough on the computer to create those virtual computers, those virtual machines.
Oh, thank you. It's a bit unfair to throwing into it. It's a big question. I realize that, but I have another one, which is which is perhaps not as easy to answer either, but it's a bit more of opinion. It's regarding the requirements for journals to have the data and raw data published. What is the appetite? This goes from Joseph Chipperfield. What is the appetite to promote or introduce requirements of compliance with various biodiversity data standards, and specifically Darwin Core, I guess, and encourage this as a next step in making data fair?
So we're definitely realizing that authors they don't intuitively know what's expected of them in data archives, and I think I think some of the problems that we're seeing when we start looking in depth at the repositories, some of it's laziness, but some of it is really just authors don't know what the requirements are. And so we are moving towards providing links to Dryad that has a list of best practices, and our journal has its own list of recommendations for what should be in a data and code repository. And we're continually trying to improve the clarity of that material. So we're certainly open to that. I actually am not, I'm not deeply familiar with the Darwin Core list. And I think part of the problem is that the type of data that we encounter is so variable. We get proteomics and we get genomics and we get animal behavior experiments that are videos and simulations. And so I think a lot of it comes down to it's hard to, if we start creating a list of expectations for each kind of data, we're going to very quickly have a 100-page document that is going to be unnavigable. So we're trying to find a balance between being succinct, which is important for user friendliness, and being thorough about articulating our expectations.
Well, thank you. Are there any questions from the audience, by the way? Comments? Everyone is typing away on the Slack here. So it's good. I was curious about how, and this goes mainly out to you, Ingrid, how you have considered various field-specific data standards in your work, and how that would relate to the verification of data.
Thank you, Anders. That's a big question, and our report covers all types of research, so we couldn't really go into detail. So we talk about it at a top level. Data should be fair as possible. For some fields of research, like your fields of research, that means really good metadata, ontologies, and so on. For others, it might just be thinking about, do I have a file format someone else can open at all? Do I have at least the Dublin Core metadata? So it really depends. And I think it's really important when we work on good infrastructures, we have the field-specific infrastructures as the guiding torches, so to speak, to see, like Elixir, you've come far. How can other research communities learn from that? That's kind of the best answer I have. It's hard to, yeah.
Thank you. So see that Alan, you posted a question to Dan here in chat. Do you want to take it in person? Regarding ensurement of data archiving? I have to see the... Or whatever you choose, anyway. Don't have it.
Hi, I'm Alan Nilsen. Thank you for a great talk. Yeah, but the question I think you're referring to, Anders, I think we have already discussed that quite well. So the question was for Dan, yeah, what would be your suggestion to ensure that the data that is archived with the journal papers are actually enough for reproducing the results and being reusable? But I think we have discussed that quite extensively, so I think, unless you want to add something, maybe we move on to next question.
Okay, That's fine. So I am.
I guess maybe I will say one thing to that, which is there are different ways of having data that's reproducible. Let's say somebody's doing a behavior experiment and you're recording how long it takes spiders to respond to a stimulus. You could provide an Excel file or a CSV file with the wait time until the spiders respond, or you can upload the raw videos of the behavior trials. Ideally, I guess I'd say both, but our standard right now is very much focused on the CSV or Excel file that contains the numeric data. Uploading raw videos and raw photos and things like that are much more data intensive, and they're not standard because there's the viewer would have to analyze each video to recreate the numbers. But actually, those videos are far more reliable as a record of what happened for detecting fraud, for example.
Thank you.
Could I maybe comment?
Yes, definitely.
So you also kind of touched upon another interesting topic, which is the methods we use and how we share those methods and the documentations and our protocols and so on. And for something to really be reproducible, especially in the lab or in the field, you really have to understand what was actually done. And as you touched upon, when you share the numeric data, when you share your data maybe 2 or 3 or 4 levels down in your pipeline, then it's really hard to reproduce or to understand what actually was done. So I think we should really think about the broad output of research results.
That's a very good question, and I think especially the move by journals for shorter and shorter papers, and I'm looking at ecology letters here, and of course Nature Science, that transition towards very short papers has encouraged authors to be very succinct about their methods, which does not help understanding. There is a very interesting initiative, if you're not familiar with protocols.io. This is a web service where it's sort of like Dryad, but for a detailed repository of your methods that are version controlled. And it's fantastic, especially for, let's say my lab is doing microbiome experiments. We detail our lab methods so well that somebody could simply read our files and do exactly what we do in the laboratory. It's at a level of depth and detail far beyond what anyone would bother to write in a methods for manuscript, but it's actually essential if you're going to recreate a result. And so we do encourage protocols.io, but we don't currently require it because it is a fee-driven service. But it's a version-controlled protocols document that can be private to your lab or public for anyone to see and adopt. And I do recommend people take a look at this. It's a very interesting initiative.
Thank you. I got a comment or question or something coming up from Dag Andresen here.
Yeah, Doug, it's for Dan and Ingrid regarding this reproducibility and the more broader assessment of. So I'm thinking, are we ready to consider maybe a shift of the publication process to physics is using preprints and post peer review, so that maybe you let the peer review process take some time. Rukai also posted something that she's shocked that review of the data is not part of the process, and maybe you could then have the paper being reviewed a little bit longer and maybe more dynamically through time. Are we maybe ready to maybe consider to embrace a larger shift in these processes?
Yes, you want to answer first, Dan?
Yeah, I mean, that's a very deep question. And I am not enthusiastic about post-publication peer review. And I'm sure we could spend hours debating this issue, and articulating my concerns. I think my concerns come from a couple of angles. One is that the few times I've tried to deal with bioRxiv as a mechanism for getting comments from people, I've gotten nothing or next to nothing or very cursory comments. And I think fundamentally journals play a very central role in recruiting reviewers who are chosen by editors on the basis of their expertise. Editors also play a very crucial role, and journals play a very crucial role in things like retraction. So if everything's just on a preprint server with comments, the process of retraction and launching formal investigations into fraud requires an institutional arrangement and a decision-making hierarchy that isn't, I think, well reflected in the proposals that I've seen for post-publication peer review. At the American Naturalist, we often get very detailed comments from reviewers, from associate editors, even editors put a great deal of comments into individual papers. And I tend to find that the intensity of commentary and the depth that people go depends on the journal they're reviewing for. And I think that there's a sociological element here where people have psychological or sociological affiliations with institutions that increase their motivation to donate their time and effort on behalf of a journal that they like and respect, for example. And so I do think that there's a genuine feature that's added there. And then lastly, well, certainly not lastly, post-publication peer review puts flawed work, and I would say that most manuscripts have significant flaws when they first come in the door. It's very, very rare that we don't see flaws in analytical approach or study design or interpretation early on in a review process. It happens, of course, but I guess from my vantage point as editor of having rendered decision on approaching 1,300 papers at this point in my position as editor, is that the vast majority of papers are significantly changed and improved by the review process in a way that makes me very nervous about putting things out publicly. We have a phrase about best foot forward. And I think if you put in the public eye work that hasn't been reviewed yet, it's not putting your best work out. And review before publication serves a very important role in helping authors avoid embarrassment, basically. And then that becomes particularly acute in situations where, for example, in the US, we have a great deal of anti-evolution movement and creationism that tries to masquerade as science. And I think scientific journals and the peer review process plays a key legal role in distinguishing what is science and what is not science and what can be taught in classrooms.
Thank you. That was a very good ending to this session. I think we are also 2 minutes over time, and I have to introduce the coffee break now, or the crowd will probably go wild. So again, thank you very much for this session. I suggest that we give the speakers and the audience a big applause for this session, and hereby declare the coffee break for open.
Hallo, hallo. Hallo, hallo. Hallo, hallo, hallo. Hallo, hallo. Hallo. Hallo, hallo. Hallo, hallo. Hallo, hallo. 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 Hallo, hallo. Hallo, hallo. Hallo, hallo.
going to the extreme, we can think of the whole South America and aggregate all that information into the whole all the sites in South America, and that will imply that there's one event with two. Data structure point of view can be thought of child data set or children data. And effort of the inventory. And this is just an example of the work that we are doing. So we select a term name, in this case, inventory type. We create a definition. We include any comments if we think it's needed, and some examples. So as you can see, it's following the logic of Darwin Core. And now, what are our next steps in the group? So first, we need to finish the revision of all the Humble Core original terms. As I said, finish reformulating the definitions, the comments, and the examples. We will need to test this extension with real data sets. Part of the team has already developed a GB5PT for this, which is obviously not public yet. But we will need test data sets. So we are more than welcome for people to join and share their data and see if these terms and this whole framework actually works for your data. Then we'll have to go through a public review as the Darwin protocol, the Chadwick protocols proposes. In the same time, we need to develop a documentation and guidelines on how to use standard. And finally, we hope for is to ratify the Humble Core extension to Darwin as a valid standard. And the whole idea is to provide the community with a solution to share and reuse inventory data, following the FAIR principles of improving data discoverability, interoperability, and obviously reuse. And a key important thing is that we are trying to develop these standards, thinking of lowering the reporting or the capturing burden. So you might feel there's a lot of terms that the people might have to include and describe, but the hope is to kind of like lower the amount of time into the reporting of this data. So thank you, all of you, to staying and being interested in this work that we're doing. And obviously we thank the whole task group. There's a lot of people here, but basically I also wanted to thank Erlen Nielsen for inviting us to give this talk and to all of you. So thank you, and we welcome any questions.
Hey, Yanni, can I add one last quick thing? We just sort of talked about having a chance to sort of double down on a couple of comments on the Humble extension.
Yes.
And I'll just add one last really quick thing. That was awesome, Yanni. Thank you. That was really great. So one reason why we've been so focused on making sure that the Humboldt extension goes through a formal ratification process with the organization called TADWIG is because the best case scenario that I think everyone can see is that we could use the existing tools that are available for publishing data that have already been developed by GBIF and TADWIG. So there's already a really great set of tools to mobilize incidental records. And so being able to leverage an extension where you can link all of the existing kind of flat data on the record side, the report of what species were found, with the inventory metadata means that we don't have to recreate infrastructure that already exists for making those kinds of data available broadly to the community. So hopefully this extension serves a real purpose in being able to leverage the existing tools we have and keep everything, all of our kinds of data that we have, whether it's incidental records or inventory data, all in a joint infrastructure that exists globally. So that's the last thing I wanted to add, and I'll just leave it there for now. Thanks.
Thank you, Janina and Robert. Due to the technical problems, I think we unfortunately just have to move on, but thank you for a very interesting and inspiring presentation, or both of you. I'm not really sure, is it Diana Bowler who's the next speaker? So I guess she needs...
I think
Hello.
I'm next.
Hello, Diana. Okay. Then you can start your presentation, Diana. I guess you have a sort of an opening slide with some information about yourself.
Yeah, can you see it now?
Yes, now we can see you.
Yeah.
So,
okay, brilliant.
yeah, I think you can introduce yourself, Diana. So just go ahead and
yeah, thanks a lot for the invite to join today. So I was actually based in Nina a couple of years ago now, or maybe longer, I can't remember. But now I'm based at the IDIF in Germany. And today I'll present some of my experiences of working with species occurrence record databases, so trying to extract biological information from these messy kinds of data. And I think my take-home message is going to be that the challenges that I've had have been rather more on not so much with getting the data. People quite happily share data with me. It's getting the metadata from these people that always makes things more difficult. So yeah, I'm working specifically on a project called ESMON, which has the long-term goal of describing how species populations have been changing through time in Germany. And we're avoiding birds and butterflies because they're easy. They've been done to some extent in Germany. We're going for the more kind of difficult groups, so different insect groups and frogs and stuff like that. So Germany is quite typical of most countries in Europe in that there's a few national monitoring schemes, but they're only for select taxa. For most taxa, there's no single coordinator scheme, rather there's more local data collection happening across the country. So different local groups are collecting data independently. And often in Germany, this is centered around the federal states. So these data are challenging to analyze because they're collected without any uniform protocol. So in SMON, we have kind of two challenges. One is just simply get all this data together, because so far it's just been scattered, and only just last year did they establish a new national monitoring center. Before that, everything was federal. And then the second problem we had was, analyzing this data once we'd compiled it. So we're working with natural history societies mostly, because they collect tons of data that often isn't available or it isn't shared yet in some of the databases like GBIF that we commonly use. So these data are just on people's computers. They're in databases of the conservation agencies, and the culture of open science is really yet to penetrate these groups. They still need some convincing on why they should be sharing their data. But we're trying to do that. So we're trying to show them that they can work with us. We will do sensible things with their data. We're not going to go off and do crazy things. And we're hopefully in the long run we'll be moving towards getting this data actually open access, but it's not there just yet. So on the left is all the people that I've been working with. So all these, mostly Natural History Society members. And on the right is just showing you some of the details of the database that I've compiled on dragonflies. So dragonflies was quite a good case study for us because there wasn't a single scheme for dragonflies, but there is a lot of data collection nonetheless, more locally. So by compiling this national data, by compiling data from different people across the whole of the country, we've been able to pretty much get a national database. So this is just showing you the coverage of records. And there's some really amazing people out there. So this higher density region of records here is pretty much down to one man, Bernd Trocker, who loves his dragonflies and in his spare time collects all this data. So I don't want to say too much about the data sharing side of things. I want to say more today about the challenges of the analysis of the data. But I thought I should just mention that there is a project in Germany called NFDI for biodiversity. It's just started like the last year. I'm just peripherally involved in it. But NFDI has an aim of developing tools, workflows so that people can use to make it easier to share their data. So to foster kind of fair data. And the Dragonfly Natural History Society and many of the other natural history societies are case studies for NFDI. So we're working with them to do questionnaires, surveys to assess what do they need from us to help them make their data more fair. So bear in mind, these people collect data already in their spare time. It's a lot to also ask them to now also develop ways to make their data more shareable as well in their spare time. So we're trying to work out how we can support them in that, so some capacity building there. But like I said, I really want to talk about the analysis today, the challenges of the analysis and metadata issues. So because this data hasn't been collected with a single protocol, it's a database of heterogeneous data. We're using a hierarchical model to analyze it, an occupancy detection model. And these are quite commonly used now these days for this kind of data. And the reason why people like this kind of model for these kind of databases is because they allow for the fact that the data is generated by two processes. So the bottom layer here is supposed to represent the true ecology of the system. So what affects where our species is. So that's things like land use and climate and maybe biotic interactions. But also, the top layer represents that the data that we have is also dependent on how the true system was sampled. So we call these observational or detection processes. And that's what we have in our database. So if we want to really model this data, we have to actually think about both these data generating processes, both the observational processes and the ecological processes. So we've done this to our dragonflies. We've used occupancy detection models to that data set that I showed earlier with the map. And we can make some estimates of how species populations have changed since 1980 onwards. And I'm not going to show any detailed results. This graph just highlights a summary. So we have some increasing species and we have some decreasing one. All I want to highlight here is that when we show the species-specific results to the experts, so that all those members of the Natural History Society, at least from their point of view, the results do generally make sense. So the model does seem to be doing a reasonable job of predicting the trend of species. But we know that our model is pretty simplistic. So these occupancy detection models have various assumptions, one of which is that there's no unmodeled heterogeneity in that detection process or observational process. And we know also that when there is unmodeled heterogeneity in these models, then our occupancy estimates will be likely biased downwards. So we have good reason to suspect that our models are biased with some respect. And that's partly because the available metadata only allows us to have rather simple detection models on that observational process. So the data that I have is very simple. It's species, date, what else? There's more, but I can't remember. Oh yeah, X, Y, so some kind of geographic coordinates, and usually some recorder name, recorder ID. So from that, so all we can really do, all we can really, the only covariates we can extract are things like list length as a proxy of effort. So that's how many species someone reported on a given place and a given date. And you see there the biggest division is between people that just submit one species versus submit multiple species. And probably those people submitting just one species weren't really doing a proper survey. That was probably a really just opportunistic record. So we hope that list length will at least model some of that variation in whether people were doing a true survey or not. We can also use this very simple data to try and generate proxies of the observer expertise. So we look at the total number of records that somebody submitted. And hopefully if we make the assumption that the more records they submit, the better they are. Maybe not always the case, but hopefully it is. And then we can try and also use that as a model for some other observer variation. But this is really simple. You know, we know these databases are super messy, and here I'm just mentioning a couple of covariates that we're trying to model that with. So what else might we think of? What other metadata we need, or what other information might we need to really make best use of these species occurrence records? So there's various ways that people might be collecting data differently. Their sampling design might differ. So what habitats are they going to? Even locally, what are they looking on the ground, or are they looking higher up? Protocols, so do they have a method like a transect or a line or a point count? Do they have a target list or not? Are they just recording anything? Survey effort, obviously, and something about ability, so taxonomic ID skills and general experience. So they're the ways we can hypothesize people vary in how they collect data. To try and better understand this, I did a survey last year where where we asked people specifically how they collected data. So we had about 15 questions of people from these natural history societies. So yeah, 900 people. And I show here just some of the answers to a few of the questions. So the top left one here shows what people said about what proportion of their records came from a planned search, where they went out intentionally looking for species, versus just opportunistic observations they saw. So we have a bit of a, well, first thing to notice that most people do some combination of these two types of surveys, but the most common was this one here. So most of our respondents did a lot of active searching, but at least some of their data was entirely opportunistic. And then these B and C shows that which kinds of species people report depends on what kind of survey they were doing. So if someone's doing an active search and they go out with the intention of looking for species, then they were likely to tick this box here saying that they recorded all the species that they saw. That's what this purple means here. Whereas lower left, if they were just reporting something opportunistically without any planning behind it, that opportunistic observation was probably triggered by seeing a rare species or seeing a species that they thought was interesting. So yeah, there were lots of questions. I'm not going to go through all of them, but I just show you here. Yeah, it summarizes really all how the responses to all the questions were related to each other. So if you, each of these blobs, each of these colors refers to a different question that we asked them. So these blue blobs here are about the species seen during active searches. The yellow is about the locations, and the lines represent correlations among the responses. So for instance, let me find one, it's a bit hard. Yeah, I think so. This activity, sorry, not for a while. Like frequency of activity, so how often they were looking for species was related to what kind of habitats they went to, for instance. So the idea is that if there were lots of correlations here, so if people were correlated in how they collected data or where they went, then in theory we could simplify that variation down to just a few metadata variables, because if they were correlated, then a few metadata covariates would characterize how they collect data. But actually you see that there's not a lot of lines here. There's whole areas where there's just no connections between the different responses. So people are really variable, which suggests, unfortunately, that we do need detailed metadata if we want to understand how they collect data. That said, if I had a wish list for the metadata that I would want for my data, it's this. And actually, it's very consistent with what you heard in the previous talk, which is helpful. So really, the top one is really just knowing whether an observation comes from a complete checklist or not. So this is all presence-only data. People only bother to write down when they saw something. But depending on how they did their survey, you can infer zeros of what they didn't see. So if they did a complete checklist, if they reported everything that they saw, then you can infer zeros for all the other species. But you have to always remember that a complete checklist can only be defined with respect to some target community. So were they looking for any bird species or any frog species, or probably wasn't any organism, because that would be a very long survey. And then if people have ticked that box, if there is metadata on this complete checklist, then we can infer the zeros, and we change our data from presence only to presence absence, which is obviously really useful for trying to separate out spatial biases. A second wish list for me is regularized recorder IDs. So like I said earlier, I do often have some column in my data sheets which is recorder ID, or in I think GBIF it's recorded by. But these columns are pretty messy. So you see recorder names like John, like who's John, or just the initial, and sometimes it's groups. It's quite hard to make use of these messy recorder names without spending a long time doing data cleaning. So that's on my wish list. and obviously something about effort. So what kind of survey and how long. And I think with these 3 pieces of information, we can go a long way to modeling a lot of that variation. And these 3 pieces of information are actually already collected by some databases. So eBird is really, for me, in the lead when it comes to this, because they already collect all this information. And you can see how valuable it's been, all the really cool stuff that eBird are doing. And I think that also highlights that asking people for more metadata doesn't necessarily, shouldn't slow down the participation of people. I think that's often one concern when it comes to citizen science anyhow, that if you ask people for very detailed information, then they're less likely to even bother in the first place. But you see with eBird, people quite happily will provide this information if you ask them in a nice way. So yeah, for me, that the problem isn't really sharing, although that still is an issue. It's sharing of metadata that enables analysis. And once we have this metadata, then it doesn't really matter that the databases are heterogeneous. You know, you can model that heterogeneity in the model if you have the covariates and the metadata to describe those covariates. And there is a tendency to assume that these databases are just generally low quality, when it isn't. It's just that we don't know the quality of the data sets within, or the data points within. So yeah, I think we had a little mention earlier from integrated models, how there's a kind of new line of models that people are developing, which explicitly would allow you to model different streams of data, like presence only and presence absence in the same framework, which would fit with this greater availability of metadata. Yeah, embrace heterogeneity. Don't be afraid of it. We just need metadata. So I will end that. And if anyone has any questions, I'll be hanging around the Slack, I guess.
Thank you, Diana. That's a very for an excellent talk and an excellent suggestion. Oh, I hope. Can you still hear us, Diana?
Are you talking to me?
Yeah, you can still hear us. Yes.
Yeah, just about.
Yeah, but thank you for offering to answer questions in Slack. I see that there's a high activity there. So I think we need to move on to our final speaker here, because of some technical issues earlier today, to keep up the pace. So please just post questions for Diana in Slack. Then it's Ivar.
Hello. Hello. Okay. Thank you for inviting me to talk a bit about my experiences as a researcher. And I think you will see throughout my talk that I am not an expert on what you are experts on. But that's a bit the point of why I'm talking, I think. I will talk a bit about my experiences as being involved in some large or long-term integrated projects, which is a word. Integrated is a word that is very popular these days. And it is popular for several reasons. One reason is that we are asked to be multidisciplinary, to have large integrated research project, which means that we have to bring together different disciplines to meet the challenges for the society, for instance, or to bring new knowledge. Also, biological research has become more integrated. Ecoevolution is one example. You have ecophysiology, or you have demographic evolution, ecology, or whatever. You just put together two words, and then you have a new discipline that is excellent. So to be both relevant for the society and to be in the research forefront, we have to be, our research must be integrated. And I think that's a good thing, but it comes with some challenges when it comes to data. Traditionally, if you have a system A and a system B, or a system can be different disciplines or different taxonomic systems or whatever, you have this sort of separate workflow, and this workflow or pipelines or whatever you call it, it may be incomplete, it may be too many steps, but at least this is a bit how you traditionally did it. You had for one system, one workflow, and for another system, another workflow, and you analyzed it separately. You collect the data and you processed and all this. But now, at some point, you have to integrate. And what most often has to be done is that you, at the end of these two pathways or pipelines or whatever, you do an integrated interpretation of your results. Then you can ask how integrated is that? I don't think that is very integrated way of doing it. But there is very many different ways to have these pathways to be integrated, and it's just your imagination that limits this, or the number of systems and combinations of arrows. I will show you. I'm not sure what that is there. Two projects where I will, where we have these different pathways of different systems. Because this workflow will depend on the project, I think. Ideally, it should be planned in advance, which is not always the case, and it will be changed. That's the only thing I'm sure of. Something occurs that make you have to change how you do this. So the two projects I will talk about is the Vega Mus project, which I think many of you knows, and the project is called Ecosystem Trøndelag, which maybe not so many know. And both projects are close collaboration between Nina and NTNU, and it has experts within different disciplines at the different parts of the workflow, all the way from sampling to interpretation. There are a lot of different people involved. The Vega Mus project, it started already in 1988, so it's obvious that I have not been involved in it from the start. It has had different periods where different research question has been addressed. It started like perhaps more classic ecology, but then it moved into evolution and population genetics, but management and harvesting has always been central. And the interaction between these three factors has always been a key question. And it has, of course, been adjusted when new technology arises, and you get new opportunities to analyze data or to get new information from old data. There are basically three main sources of information. You have GPS colors, you have like classical body measures, and you have tissue samples. And much of this is sort of collected or initiated at the same time, because you go out and you immobilize an individual, you put on a GPS collar, you take some tissue samples, and you take body measures. So we can sort of say that that the sampling, initial sampling, is common. But then these three main data goes into different pathways. So the GPS data goes into a machinery of storing big GPS coordinates, and has its own way of processing and cleaning the data and storing it. The tissue samples is used for mainly for extracting DNA and doing genetic analysis, and goes into a completely different machinery of analysis that I know not too much about. But the output is, for instance, a pedigree like this with relatedness, or it can be genetic structure, or it can be heterozygosity. It's a lot of different measures that comes out with this from a process. And then you have the ecological data, which is a bit... I didn't have an Excel sheet open, so I just took this, but it's like the age and sex distribution. It can be body mass changes or whatever, which goes into a different database. So I think the pathway for this project is mainly like this, that you have common sampling, which is the marking on the individuals, and then the data goes in different pathways, and then it goes together when we start analyzing it and interpret it. It looks fairly simple, but of course it's more complex because you have a lot of different sources of data that also go in. For instance, the hunting statistics, observations of marked individuals and their reproductive status. Car accident is an important information for ungulates, for instance. And then we have also started to collect hunters' data, give them a GPS and see where they go and when they encounter moose, which has its own challenges with respect to, for instance, how can you use data from humans. And all this data at the moment, as far as I know, is maintained because of 3 persons at NINA. They are doing their job, and they are central both for collecting the data, for storing the data, but also for getting out the data. So this shows how challenging it can be to work with this type of data. For instance, the genetic data, there is a pathway that it sort of follows, but then there are new tools, a new way of analyzing the data. It's new. You have to go in and maybe extract some DNA again. So it's back and forth, which means that you have to be flexible. You cannot. You have to have a dynamic way of storing, analyzing, processing the data. I will talk a bit about this project. This is quite different. Here is the aim to better understand the variation in biodiversity in Trøndelag at different spatial scales. And the main reason is the huge changes that we observe in Trøndelag and in all of Norway, basically, where the landscape is changing at a rate that I don't think we have ever experienced before, due to infrastructure development or other constructions or destruction of nature. So how will this affect biodiversity, and what areas can we afford to lose, sort of? What is a very important area that we cannot afford to lose if we want to stop the biodiversity crisis? And then we see that we have this type of data. It's from the GBEF database. A few billion, is it so? Data points. So there should be data to address these questions. But as it has been pointed on, if you zoom in a bit, even if Norway and Trøndelag is sort of a hotspot of biodiversity data, it's quite scarce when you go outside the cities, or if you try different habitat type. And Anders showed this figure. It's also quite poor for many of the taxa. So the problem is that we lack data from many taxa and systematically sampled from the same location at the same time. If you want to say something about how important the habitat is, you need surveys from the same location at the same time. So we need more biodiversity data. We cannot rely just on the 2 billion observations in GBIF. As an example, this Storheia wind farm, which currently is the largest in Europe. We'll see how long that will last if it disappears. The available biodiversity data, the last 20 years, it's 11 observations per year within that area. And that is the type of data that authorities used to find out where can we afford to put out windmills in Norway. So keep on sampling data. That's my main point. So that's what we did. We set out 3 areas in Trøndelag where we distributed some sampling areas in different habitat types. And we reduced well-established methods for doing surveys of vegetation, insect, and birds. And if you remember a few slides ago, that's not filling the knowledge gaps, but we are a bit faithful to our study organisms, so we do what we are good at. But then we get these 3 pipelines for the 3 different taxa, because they are sampled very differently. Vegetation, it's surveys with squares where you have detailed information about the plants, or the coverage of plants, because counting individuals in plants is not straightforward. For insects, you have a different way of sampling with the traps, and you have enormous amount of data that maybe are sent to a lab for barcoding. For birds, you do transects, and you have to multiple times a year, and you have to do some sort of decision of how many territorials or nesting attempts that are there. So there is 3 different ways from sampling to data and maybe storing those, and then you have to do the processing and get some usable data that we actually can use for our analysis, which also have to be stored, and then you start analyzing and interpretation. So I think that this is the way forward for this type of data, that you have to have 3 different pathways before you have storing the usable data. But you also, as was mentioned, you need to store the raw data, and that has to be done in, I think, from what I've seen so far, in different systems. But we have not come that far. But Living Norway will be very central for this work. And maybe even not storing the final data can be done, that we have to look at. But it's only in the last part, or the first part, the planning part, and the last part that you have true integration in this type of project, I would say. Then I will focus on a bit different challenge, which is this language barriers. And I struggle. I have maybe it's something with personal interest in the topic or whatever, but there are so many words and so many acronyms. And this data management and data sharing and archiving, it has its own language that I am not yet familiar with. So that means that I have some challenges to follow it. It takes time to learn this language, I think. New words pops up, and acronyms, they don't help. Sorry for that. And sometimes I feel a bit like this fellow to the right here. You have a very enthusiastic choir that it seems so simple for you that know the language, but for me, I just keep forgetting it every time I talk to you. So that is a challenge for me as a researcher that is not really dedicated to this on a daily basis. So some personal thoughts to conclude this. So we have different type of expert involved in this project in different tasks. For instance, sampling, monitoring vegetation. I couldn't do that. I can't enough about that. Insect identification or DNA analysis. So different parts of the project have their set of experts doing the job. And I think that we also need experts to do the data management, data archiving, data sharing, and standardization. I think maybe it's a bit too much to expect that researchers also should do this. I will never go into a lab and do DNA extraction. That's not my background. And it's also not my background to standardize data. Maybe it's as simple as you have described today, some of you, but that would help a lot. But still, I think we have to educate and we have to hire these experts. For instance, the Department of Biology, they have 23 technicians. Not all of them permanent, but at least in the list of people, there are 23 technicians. I know of one of them that work with data management, databases, and archiving. He's not permanent, and that's not his full-time position. If you can have people that have responsible for one electron microscope, I think you also can have people that are responsible for the data that are generated at the department. And do not expect researchers to understand your language. That's for true. And fair is not the same as free. I have the impression that some people a bit high up in the system thinks that it's just to do it. But I think it costs. It requires resources to do this job. So we need these resources to be allocated. And that, I think, has to be realized from among people above our level. And we need the people to do the job for us, or for me at least. So that's it from me.
I think you can just wrap up, Alan. There's no need for me to drag out the time here.
Yeah. Hi again. I will not spend much time on this, but I think it's nice to say a few words at the end of the day. First of all, I would like to thank you all for spending the day with us, both those that joined us remotely and those of you that came to Trondheim and joined us at the Ninahuset. I think it has been a great day. At least for me, it has been a great day. I enjoyed all of the talks, all of the presentations, and I will take the opportunity again to thank all the presenters and also the rest of you that contributed in different ways, asking questions, joining discussions, and so on, both in person and on Slack. I would also like to thank those that help us to ensure that this runs smoothly, at least mainly smoothly. We had some challenges at the beginning of session 3, but that was solved relatively promptly, and so thanks a lot for those of you that helped us making this the technical part of this event. So I hope to see all of you tomorrow again. We start tomorrow at 9 o'clock Norwegian time, 9 a.m. Norwegian time, for the first workshop on reproducible workflows. And then after lunch tomorrow, we will have this open working meeting where we discuss various issues. And I will again remind you, that those that are joining us remotely, that if you want to join this second workshop, please let us know so we can arrange with the video meeting for that. The next thing that will happen here now is that we will have a dinner at 6 o'clock Norwegian time, so see you all then. Thank you.